It is our privilege to welcome you all to the Industry and Innovation session of the Sustainable Biotechnology and Bio-Innovation Conference. This session brings together visionary leaders from industry, startups, and translational research who are actively transforming scientific discoveries into sustainable solutions. Today, we explore how biotechnology is moving beyond the laboratory into scalable technologies, green manufacturing, circular bioeconomy models, and real world impact. Innovation is no longer optional, it is essential. From bio-based materials and renewable bioprocessing to climate-resilient technologies and waste-to-value systems, sustainable biotechnology is redefining how we design, produce, and live responsibly. We are excited to hear insights from pioneers who are bridging the gap between research, commercialization, and societal impact. Now, it is our honor to introduce our esteemed session chairs who bring a wealth of expertise and leadership in both academia and industry. We have our first session chair, Dr. Garima Agarwal. She's a seasoned academician and an industry expert with over 20 years of experience across industry, academia, and research. She holds a PhD and MPhil in management, along with advanced qualifications in marketing, export management, and education. Her research on the contribution and potential of small-scale industries in India's export sector reflects her strong focus on entrepreneurship and economic development. A certified global jury from Vadvani Foundation and innovation ambassador of IIC, MHRD, Government of India, she has worked extensively with the Ministry of MSME, Nice Word, NSDC, NYKS, and PMEGD, training defense personas, youth, and aspiring entrepreneurs. She has also been associated with Jamia Millia Islamia and currently heads the Amity Center for Entrepreneurship Development at Amity University, mentoring students and leading government-sponsored initiatives. Recently, she collaborated with the Government of India and Vietnam to strengthen entrepreneurial ecosystems and is a recipient of the UP Nari Shakti Award 2025. Please put your hands together to welcome her. Ma'am, you're requested to kindly grace the dais.
Our second chairperson is Dr. Santosh Kumar Mishra. Dr. Santosh Kumar Mishra is currently working as Associate Professor and Head in the Department of Life Sciences, Sharda University, Greater Noida. Dr. Mishra has completed his MTech in Biotechnology from the Institute of Engineering and Technology, Lucknow, UP, and his PhD research work from Indian Institute of Technology, Roorkee. He has more than 21 years of teaching, research, and academic administration experience. His area of research are bioprocessing and downstreaming, industrial biotechnology, phytochemical analysis, and computational biology. He has published more than 30 research articles in international peer-reviewed journals and also presented his work at various national and international conferences. Dr. Mishra has also edited 3 books and published more than 15 book chapters with prestigious publications. He has also served as editorial board member and reviewer of prestigious international journals. He is a life member of BRSI, IFERD, and SBBS. Dr. Mishra is also recipient of Excellence in Teaching Award by Society for Bioinformatics and Biological Sciences and Excellence in Academic Leadership by IFERD. His research group is presently working in the area of microbial enzyme production and process optimization using low-cost agro-industrial residues. I request our distinguished guest, Dr. Santosh Kumar Mishra, to please take his seat on the dais.
Please put your hands together and welcome him. We are truly honored to have them guiding this session. With that, I request our respected session chairs to take over the proceedings and lead us through this exciting and impactful session. Thank you, everyone, and I wish everyone an inspiring session ahead.
So a very good afternoon to all of you present here. So after the lunch, the session used to be a difficult one. So trust me, we are not going to bore you because the topic is very much relevant. So whenever we are in the societies, So we have to see what the problem statements are and how we are going to sort out the issues of the society, not today, in the coming 5 years. And this is a problem we all are facing, like temperature change, climate change, lots of things are happening. In fact, the water levels are increasing. So we all are realizing that problems are existing. But how to identify those solutions which are going to help out for the upcoming generations? Okay, so today's session, obviously, it's a very nice session which is given by this conference people. They are talking about from information to innovations. We all are having many informations available with us. Like I used to give example of the movies to all my students, reason being ki you can easily connect. There was a movie of Salman Khan. He said, if you want to change something, you have to train 5 people. Those 5 people are going to create a new chain of next 5 people. So same way it happens ki when the information is there, discuss among people, identify how can we add value to it. So that's why today's session is very much important for us. Information to innovations, how biotechnology can change the world in the coming time. And trust me, upcoming time is a part of AI. AI is going to be the tool of us, and we are going to utilize those technology for the development of our areas, geographies, and the hemisphere where we live. Over to you, sir.
To all of you. And the theme of this conference is sustainable biotechnology and bio-innovations. So I think when we are publishing research paper, we are adding the keyword that is related to sustainable. But I think in the nature, there are 4 order, that is a material order, plant order, bio order, and human order. Except to human order, there is no knowledge to other order. So therefore, human developed and emerged significantly time to time, and at present we are here due to evolution. But we must realize what is the role of other 3 order, like material order, plant order, and bio order, because their role is definite. So if we are having knowledge, so this is our responsibility. We should not damage and destroy the rest 3 order, and we should not also create problem to rest 3 orders. So we must keep all these points whenever we are conducting any research or doing any act. We must think about it. And my next point is, since there is a lot of research going in the area of biotechnology and life sciences, but we must realize what is the real problem of our country, what is the real problem of our society. It is not always we must do research in the area of cancer biology or molecular biology. These fields are also very important, but we must think what is the real issues of our country, because agriculture waste management, this is the biggest problem. Pollution is a very big problem in our country. So we must think what is the current issues in our country, and we must focus our research in that specific areas. This is my message to all of you, and hopefully in this session we will listen some fantastic lectures and presentations, and we will increase our knowledge. Thank you.
So now we would like to invite Dr. Richi V. Mahajan. So I will like to introduce him because we are very uh, delighted, happy, and it's a pleasure also to have you, sir. Sir is as a working as a scientist D at the Department of Biotechnology (DBT), Ministry of Science and Technology, Government of India. He specialized in the microbiology and biotechnology with key contribution to biological data management, genomics, bioinformatics infrastructure. A Delhi University alumnus, Dr. Mahajan has several publication and book chapters to his credit. He has played a significant role in advancing DBT's flagship programs like Genome India, UMMID program on screening of rare and genetic diseases, artificial intelligence, the biotechnology information system networks, biotech pride guidelines, IBDC, FEED protocols, and BIRAC. His efforts promote secure data exchange, research reproductivity, and innovation, supporting India's bioinformatics ecosystem and Viksit Bharat 2047 vision. We welcome you, sir, at the Amity.
Yeah, good afternoon, everyone, and welcome to the session after lunch. Thank you, Dr. Garima and Dr. Mishra, for the kind introduction. One thing she missed out was that I also have roots from Guru Nanak Dev University, and my honorable professor is here with me. So it's more like a viva voce rather than a presentation here. What I would say, because this is kind of going back to 2009, where I was actually a student and he was a professor taking a viva voce. So it's actually an honor presenting in front of him, Professor B. A round of applause for that. So I've been informed that the session actually proceeds after lunch, and I know how sessions can be. At your age, actually, I used to go to sessions or conferences only for free lunches and goodies. But I'll admit in front of but I'll try to make a story out of it. Probably if you find the story lucrative and interesting, you can ask me the question. Otherwise, there's still something to relate to you people. Okay, this is quite buzzing. So talking about information to innovation, right? And we have been, just let me give you a brief about India's bioeconomy. So we have been a rising bioeconomy. We contribute to around 4 to 5% of our bioeconomy, and the mission is to reach the goal of 300 billion. Right now, we are approximately this figure is 150 billion. Right now, if you ask me, in 2026, it's 165 billion, and we aim to reach at around 300 US dollar bioeconomy. And how are we going to do that? We have skilled manpower. We have affordable markets. We have world class infrastructure. We have had examples of vaccine drive in COVID. We could actually vaccinate more than 175,000 doses. 175 million doses actually. So all these things, and again, government has been pushing about the ease of doing business, as well as NITI Aayog has issued about ease of doing science. So all these factors are actually going to make India 300 billion bioeconomy. These are just some of the stats which you can Google. That we are third largest biotech destination in Asia Pacific and second highest number of years. So we are basically called manufacturers, or what I must say, pharmacy of the world, where we are actually exporting to 190 countries the drugs. So we are rightly, and just to give you a heads up, every second child in the world actually takes Indian made vaccine. So that's how it has been, and Department of Biotechnology has been venturing into various domains like health, agriculture, energy, environment, human resource, and collaborations as well. Collaborations, when I say, I mean international collaborations. Today, I'm going to actually, okay, again, as I said, right now it is valued at 165 billion, and the total GDP contribution is 4 to 5%, and at the annual growth rate of 9.75%. So we intend to be, when there are three stages of revolution. Initially, it was industrial revolution, then there was a mechanical revolution, then there was an IT revolution. Now there's a biologization or biological revolution which is going on. And this is what we need to harness and harvest. And so that our economy, actually bioeconomy, can reach to 10 to 15% of our total GDP, so that we could actually evade that LMIS status to a prosperous country. In line with this, Honorable Union Cabinet, actually chaired by Honorable Prime Minister Narendra Modi, launched the Bio-E3 policy, and a clarion call was given from Red Fort also in the last speech, where he actually urged the youth of India to actually dwell into biotechnology as well as biological innovation, so that we can grow and develop into a prosperous nation. Under this policy, we have identified 6 domains: bio-based chemicals, functional foods, precision medicine, climate resilient agriculture, carbon capture and futuristic and space research. You all must be aware of Subhanshu Mishra, who was there in the last International Space Station mission. Three innovations from Department of Biotechnology developed at ICGB. They were carried out by Subhanshu over there, and soon, and we are still in process of accumulating the results. One of our students from ICGB, she was stationed at NASA for the purpose, and probably will be having very soon the results about it. And it was more about life in space. Coming back to, I'll start from the omics thing, like, because this represents a paradigm shift in biological sciences, and scientists can comprehensively analyze the biological systems and to the scales which were previously, and with the advent of technology as well, which were previously unimaginable. And all this is to actually provide the health and food security. If you remember, probably in 1966 or somewhere, there was a Bengal famine, and much of the population was being wiped out, and the pressing challenge to the country was feeding the people, where the then Prime Minister of India, Sri Lal Bahadur Shastri, he urged the people, I'm not sure how many of you are aware of this or not, but he urged the people to skip one-time meal, right? And so that everybody can be fed. Now in 2020 or 22, I read an article that right now the problem of India is obesity. So you see that from the mean to a different kind of problem, obesity, we have grown so much, and that has been the power of green revolution, the milk revolution, all those things. So we need to harness these powers of biological sciences for addressing the pressing needs of health and food security. Coming to like precision medicine's topic, like each size does not fit all. Probably a medicine which is responding to me and 80% may respond to you, slightly lesser, probably 70% or 60%. Giving you an example that a change in one amino acid can actually change the whole context of the treatment, right? So variations actually lead to observable changes in phenotypes, but they also actually affect the disease metabolism, the disease progression and drug metabolism in the human beings. What happens today is that supposedly there is a patient. This is a typical example I've taken for NFLD, and there's a patient and he goes to a doctor. There's a series of tests run for him. He reviews the symptom, and probably we are treating each patient symptomatically. And this is what we call as managing the disease and not predicting the disease. What will be the future? Future will be the biology-driven healthcare. So what we're going to do is we're going to create multi-omics twins. This proposal actually is already in Ministry of Finance. We received in principle approval for it. We are in discussions with them. So we produce multi-omics twins, which we apply to the pathway level dynamics, and in a few years' time, probably each one of will be having a personalized health plan. Talking about the digital twin, so Houston, we have a problem. This was actually the statement, very famous statement from Apollo 13, which was probably when I was used to study in Delhi University or Guru Nanak Dev University. I used to call my dad, I have a problem, and he used to ask me, these people were 2 lakh kilometers away. And they said that we have a problem. There was an oxygen cylinder burst in the Apollo 13. These lives were at stake. NASA took around 13 hours to actually solve this problem. And there was the first digital twin, which was back in 1979 or 69, which was, kind of carved by NASA. From there, as I mentioned, 1969, 1970. So from there, we have progressed a lot. The market has grown exponentially, and right now the digital twin market is poised at around $42 billion, but we are expecting it to be $160 billion in coming times. And these are some of the companies which are actually pioneering in these. I'll just show you some examples what this works out. So there have been a lot of clinical applications in transforming healthcare, cardiovascular diseases, surgery, pharmacy. I'll tell you what happens is, just in the second, this slide, we have a pointer or something? Okay, we don't. No worries. So this is a patient which is actually going to go, which has the case of arterial fibrillation. And this is Phillips digital twin concept. What happens is the information travels two ways. Usually what happens is when you make a model, you feed the model with the information and you come out with the solution. In this case, the information is being processed two ways. Like the information is being relayed to the system and the system is relaying the information back to the user as well. So what happens is, supposedly I'm a doctor or, a physician who is unsure where the NRM should be. He would be given best case possibilities of survival. Something you used to see in this Avenger movies, Iron Man used to do with Jarvis, right? So something like that, just to give you correlation. So you just need to see that what is the best possible, survival chance for which case, and that is how you treat a patient. So basically, you are actually not doing experiment with the patient, but the digital twin of the patient online. And once you see that there is a success rate, then you kind of go with the surgery. So that actually increases the chance of the patient. And this one actually is quite a nice one where probably, sometimes there are calcium deposits in the heart and we have a bypass surgery. And during the open heart surgery, physician is unable to relate that where to make an incision or where not. So the model is actually to walk into the heart of the patient, take an overview of what's happening, and this is based on the information, real-time information during the surgery, being relayed from patient to the system. And then you probably, and these all systems are actually available. You can YouTube them, Google them, probably. There's a very touching story of Thomas Helliker and Liam Helliker. Basically, he's an immunologist, and his son was diagnosed with a incurable disease, and he actually produced an immune digital twin of his son. Doctors did not expect Liam to survive beyond 2022. Liam is still alive and healthy. And as I said, we are venturing into other smart technology verticals. Agriculture is one of them. And these are some of the enabling technologies, Internet of Things, machine learning, and cyber physical systems and remote sensing, which enable this kind of digital twin. One thing we are trying to venture is biomanufacturing digital twins. This is something difficult to achieve. Reason being, you don't know how the biological systems are going to operate inside a closed chamber. Because usually it's not the shake flask. Shake flask is our kind of things, right? We do in the laboratory. But when you go to actual biological setups, there are closed systems, and we cannot probably interfere with the whole thing. So this is one thing which people are venturing into, but with a very less success rate. But as per the estimates, this actually comes into reality, we will have 75% of the cost reduction in the prices. So just see that in the prices of manufacturing. So just see what impact it's going to have in the coming arena. So this is what actually if I give you a concept what digital twin is actually. Supposedly I'm having a car and you also having a car. We have a same car, and when we purchase a car, we are told that we have to get the car serviced at 500 kilometers, right? That's the first thing we are told. But supposedly I'm driving that car on Janpat, and you are driving the car just at the backside of the Amity Institute, where there's hardly a road I could see today. So, do you think both our cars need the service at the same time after 500 kilometers only? Maybe, probably mine would require it at 700 kilometers because I'm driving at Janpat, and yours requires at 300 kilometers. But if there is a model which can give us a real-time information that, okay, your brake pads have, kind of worn out, or probably your plunges are out of oil, maybe you could actually, sense that probably I should get my car serviced at 300 kilometers, and it saves money for me also to getting. So that's actually the power of digital twin. But all this comes with the data. You have to have a lot of data for that. And India is an ethnic museum. We have more than 400 castes and subcastes with more than 4,600 populations all around the country, from east to west, north to south. And, we have actually a unique genetic signature. And this is mainly because of highly endogamous populations in some of the areas, as well as conservativity of marriages between the relatives, what you say, and which creates actually a distinct pool amongst the India. And the global genomics data is why I say this is that the first genomics surveillance which was carried out by Pan World was way back in 2011 or 12. 3 sites were identified from India as well. There were 1,000 samples actually throughout the world because WGS was not an easy technology back then. So 3,000 samples, three kind of sites were identified from India as well. But finally, when they were publishing the results, none of the Indian site was taken into the publication. So basically, we don't have our own genomics data. We never had our own genomics data. There was some initiative by CSIR rather regarding IndiGene project, but those was only regarding the SNP identification in some specific population, not the whole genomics data. And without our own DNA, as I said, a Western person may have a different efficacy to the drug, but we are actually preparing or manufacturing those drugs based on the genomics data of that Western person, which may not have same efficacy in me or you. Based on that, we came up with a Genome India initiative in 2020, where these three things were targeted: health, prosperity, and security. So I'll run through this probably in the coming slides. The mission was to sequence 10,000 healthy individual genomes and establish the first reference Indian genomic genetic diversity, like Indian genomic reference actually. And with this, we also have actually established a huge biobank at CBR, Center for Brain Research in ISC Bangalore, which has near to 20,000 samples. So this was, a national effort again from reactive healthcare to preventive healthcare. These are some of the stats which are easily available. I'll just run through this slide in the interest of time. So you could see that we had a team of around 20 institutes. They were all IITs, ISCs, GBU. There were teams from Northeast. There were teams from Kashmir. There were teams from IIT Madras, Mizoram University. So we actually spanned whole of the CDFD, IIT Bombay. We spanned whole of India, and we collected the population of actually initially we collected the samples from 99 different populations. But then when we were doing the statistical correlations, they turned out to be 83 populations only. So this is just the Genome India initiative in stats. As I mentioned, there were 4 sequencing centers. There were a total of 20 institutes associated, and one was RCB Faridabad with Indian Biological Data. And this was termed as an important milestone in biotechnology revolution by Honorable Prime Minister of India when he dedicated this data set to the country on 9th of January last year. Even this was highlighted by Honorable President in her Republic Day speech the same time. So just giving you a blink of what we did with this data. I'm sorry for this figure. This is 165 million now. I did not update this slide. This is 165 million genetic variations that we have found right now, and out of them 6.9 million are common variants and 1.4 million are functional variants. Now, to our surprise, we could actually curate 7 million novel variants, which was not reported earlier in any of the global data sets. Okay, so just think that they were actually inventing the medicines for us, but with a very skewed eye, or I must say, probably in their own interest, not the Indian interest. So this is general, like to understand the slide for the general public, that around 33% of the DNA changes identified were previously unreported in the global study. And many of these were associated with I haven't gone into the technical details. I have a 100-slide presentation which was presented to PMO. So I've skipped all the technical details, but I thought that this would actually give you a flavor of what Genome India we have been doing. And it has had the impact. There has been a disease linked risk profiling. affordable diagnostics are being developed and tailor-made treatments would soon be the whole of the system. Just to give you, I don't know, I mean, this is a very recent development. I'm coming from that meeting only. Initially, we had only 10,000 genomes. Now the government is actually looking into 1 million genomes, and that's going to be a boom, right? So that's going to be a humongous amount of data which probably every company, every researcher would aim at. And so we've actually got in principle approval for that from Ministry of Finance, and hopefully we'll be launching that soon. And we have been part of the genomics revolution in agriculture as well. This is one example I've quoted, which was developed in NIPGR, which is one of the DBT Institute, what these days we call BRIC Institute, which is an ethics organization of DBT. It had exceptional performance under water stress and conditions and high yielding variety and nationally approved. Now these already the farm trials have been conducted in Bulandshahr, and very soon these seeds would be actually distributed to the farmers by ICR. And apart from that, for these kind of innovations, we need to have a policy shift as well, where we came up with these guidelines on genome editing and streamlining the approval, and there has been a significant funding to support it. Now coming to the bioinformatics ecosystem of India. So, the Department of Biotechnology came into existing in 1986. Somewhere even actually I was not born that time. So that's when it came into existence, and with that only the visionary leadership that time had a vision. Sorry, that bioinformatics or IT is going to be the main key this driver for the biotechnology. That time there was a network launch, BTS network, which we call it, and that network was run for around 30 years. We had more than 160 centers in that network throughout the country, where the motive was to actually give the young minds flavor of bioinformatics. In 2020, we realized that now that flavor has been delivered, so let's delve into something more interesting. So we came out with BTS Network 2.0 and National Network Project, where the focus increased from nurturing the infrastructure as well as the human resource to research component as well, where we had a lot of collaborative research programs running in this, and that has been the national impact, capacity building, data resource development, computational tools, and all this we make it mandatory that all these are open source only. Right now we have around 52 active centers, wherein in last 3 years, 6,500 plus manpower have been trained, out of which 3,500 have been single beneficiaries, and more than 850 publications from all these centers. So you see, like, the impact has been good one. And these are the databases and the software tools developed, and the innovation metrics. So these are all figures for you may be interested in. And then we have established a lot of infrastructure in the country, and this is free to be used by any researcher throughout the country. Again, when we say Genome India, we want that data of the country should remain in the country. We don't want data to go out, because probably identifying those 7 million variations, somebody can actually devise a super bug against you, right? And probably it will be our country only, or probably another country which will be facing a COVID-like situation and not any other place. So for that kind of thing, we actually partnered with CDAC Pune, and we developed a environment called as computing environment known as integrated cloud environment, or what we call ICE. And this actually provides user dashboard and storage service as well as computing facility. So I'm not sure how many of you are aware of UK Biobank model. They don't allow you to download the data. What you have to do is you have to go in over there, you have to pay for the compute charging, and they allow you that a set of data you require to work on. You have to compute it over their systems only, over their GPUs only, and you can only download your secondary data and go back to your place. So they don't allow you. UK Biobank does not allow you to download the data, and we are actually trying to make similar arrangements. And there's some of the features of ICE. So now we have realized that probably even the research flavor has been delivered to the youth. So now the time has come where actually there should be a delivery. So now we have come up with the call known as BioGrid, which are going to be AI-powered centers where actually the output and the outcomes of the projects would be monitored. So this call is still open till 28th of February. Apart from that, we also carried out a human gut microbiome initiatives with more than 15,000 samples. 15,000 libraries and 3,000 samples. This study is again still under analysis. Yeah, so this was one study which came out with Dr. Swapnil Rane and Dr. Sandeep Sareen from Tata Institute. How much time do I have? Okay, I'm just winding it up. So this was one study which was mainly meant for images. So what happens basically is we really say that we are short of radiologists. So these are going to be the centers which actually give you a tailor-made a tailor-made analysis for the radiologist for a step one analysis so that there could be more precision in the treatment regime. I'll just skip through some of these slides. This is again one program which I've been running through the UMEED program, Unique Method for Management of Inherited Disorders, wherein we have actually established 30 Nidan Kendras and adopted 31 aspirational districts. And we are carrying out genetic testing of newborns and pregnant women. And these are as I mentioned, that biomanufacturing requires data-driven research. So we are actually making bio AI hubs where we have design, build, test, learn model probably adopted from Silicon Valley, and we are trying to emulate it over here. But important thing is why share biological data? It actually accelerates the discovery, enhance the transparency, and prevent duplication. You'll say, I did a research, why should I share it with others? But that's the reason you should share. And however, biological data presents these kind of challenges. They are vast, they are heterogeneous, and dynamic in nature. Things keep changing. According to an estimate, around 40 exabytes of data is being generated in the country, and we did not had a mechanism of sharing to it. So that's why we came up with Biotech Pride guidelines and feed protocols, which actually facilitates and enable the data sharing. It does not deal with the generation of data per se, but it actually, so if you all are aware of NCBI or EBI, so this is your Indian EBI or NCBI, and we are in talks with them. We'll be shortly part of the international coalition as well. We've received, there were 6 approvals required. I've received 5 of them. The 6th one is once they're done from MEA China mission, then probably will be part of them. And this was released by Honorable Minister last year. And this is the center I'm talking about, Indian Biological Data Center, which has huge GPU and CPU capacity. And we have made it mandatory for all DBT projects to deposit the data in this center. And these are some of the portals associated. So as you know what has been going on in global scenario, probably any day, the Honorable President may say, I'm not giving you the data from NCBI. So, that's why India should actually have control over its own data. And since it is a high value target as well, because you can change the lock or the key, but you cannot change your genetic data. So that's very crucial. So, I mean, this is what I wanted to actually tell you about. These are actually some of the slides I presented probably a few times back in front of PSA's office. So that's what I messaged her also. So this is really a race to the future, actually. And if you ask me, that we have a huge and humongous potential, and we need to actually harness this potential. So one thing I would definitely like you people to do is to visit IBDC website and see how is it different from NCBI, or how friendly or how unfriendly it is from NCBI, and probably get back to us with feedbacks that how much are you able to use it. Thank you.
Really a wonderful presentation. In fact, it is a very big eye opener for the students who are sitting here. So एक thought आता है ना, daily night 12 o'clock और getting up 4 o'clock in the morning. मैं biotech कर भी रहा हूँ तो फायदा क्या है इसका? So I hope the students are going to realize how much potential is having this area, and how can we develop, how can we go to the 1,000 genomes to the millions of the genome. So there is, जैसे कहा था ना, जामवत को अपनी शक्ति का नहीं पता था. So same way you people are at the initial stage, now you realize, and really thankful to you, sir. And we are very much privileged that the presentation which was given to the PMO, and we people are witnessing it. So thank you so much, sir. It's our really, and we are thankful to Amity that every time we find so many big giants coming in, big people coming in, and they are talking about what the government of India is willing with the vision of Viksit Bharat 2047. Thank you so much, sir. So with this, I would like to invite our guest from online session. Okay.
Thank you very much. Very interesting talk. I have one question. Like the facility you are developing so that we can do all the computation in your facility.
Yeah, so I actually did not include those slides in this. As she said, I did include some of the slides from PMO, but not all. There are some confidentiality. But now we are coming up with a national genomics data grid. So when we are talking about 1 million genome, it's going to be a humongous task, right? Like US probably is one country which has achieved that, no one else. So obviously, when we were thinking about it, we also had a plan to have there were 5 arms to it. Probably I cannot show you on slide, but there were 5 arms to it. One is the genome sequencing. Second is the Indian Biological Data Center, where you need to store information. Third arm was the AI and analytics. So probably that is one center, and that is you envisioned it somewhere in Faridabad under SPV. I think that's the information I can tell right now because it's under cabinet consideration. But there will be an analytical center which will be hosted somewhere in Faridabad, and there actually you can make in the logins. Initially for researchers, I think they are making it the provisions for free, but again, this depends on what models work out. Is it the government model or the PPP model? Depending upon that, probably the computing will be.
Like I was working in Sweden and they had this supercomputing facility.
Yeah.
Any researcher, they just have to give one
Right.
page write-up and they give us like some
that's
hours of computational power.
That's actually how we operate in Indian Biological Data Center.
Do researcher need to pay something?
No, right now not. That's what I said. Right now it's free of cost.
All right.
But they are coming up with PPP models, so I don't know what future entails.
Thank you.
One quick question. I am Dr. Manish. I have a question related to the database. You discussed that you are going to launch database, like there is one international database collaboration like NCBI, DDBJ, and EMBL, right?
So that's called INSDC.
Yeah, INSDC. So in the similar way, like you are also.
So we'll be the 4th partner in INSDC.
Yes. So that is publicly available for the person. What about the own data? I'm asking about the term own data.
Yes, that's what I'm saying, that everything is not publicly available. Only what can be given publicly, as he mentioned correctly. Some data, there are 3 types of data, which I actually skipped in the slide. There are 3 types of data. One is the open access data, one is the managed access data, and then there is no access data. So open access data is usually the structural data, which can be seen by anybody. PDBs are there. Then there's a managed access data. Supposedly this is a Genome India project. So we cannot actually give this data openly to public because there is associated metadata. कुछ population तो ऐसी थी कि 16 लोग हैं सिर्फ इन population.
Even big data, we have the NGS data also that is available.
That
we can download that.
So you see, if we give away the metadata and if कल को कोई company does something super flawless, so we would be caught, right? And this was not an easy task actually. Some of our personnel actually they had to stay in the jungles for hours to get the data. So the thing was that this falls under managed access data, and that's where you write a project. If you see, there's a data management committee. If you see that your proposal is genuine and you are a genuine person, like genuine as in you have institutional affiliation, then the data is provided to you. And then there is a no access data, which actually, as under the Government of India laws, that cannot be shared with anybody, which may lead to an upliftment or uprising. So that's something that we don't share.
Thank you so
and
much.
that's the case with NCBI and DBI as well.
Yeah. Good luck for your international collaboration also. Thank you so much.
Thank you.
Yeah, we can definitely take up more questions in the networking tea session, which will be followed after this session. Please, you're requested for that. Also, Garima ma'am, who is our chairperson, session chair for this, has to leave for an urgent meeting. So, of course, ma'am, you can definitely leave. And before she leaves, I request Dr. Devi Chaudhary ma'am and Dr. Keetan ma'am to kindly felicitate her with a memento. I request you all to please put your hands together. She said she might come back if her meeting gets over. But thank you so much, ma'am. So we'll continue with the session. So you're requested to please introduce the next speaker. The next speaker will be joining online.
To continue this session, we have the online presentation by Dr. Richa Kuthari. And Dr. Richa Kuthari is a professor and head, Department of Environmental Sciences, Central University of Jammu. She have over 20 years of teaching and research experience
I
in
request all the
energy and
students to
environmental
please
sciences.
be seated. Do not leave the hall. Please be seated.
She holds an MSc, MPhil, and PhD in energy and environment, and is a Stanford listed top 2% scientist. Her research focuses on clean energy technologies, including bioenergy, algal biofuels, waste to energy, solar-based wastewater treatment, and green hydrogen. Dr. Kothari has authored 140 plus SCI Scopus publications, 11 edited books, and 50 plus book chapters with 10,018 citation, having H index 49 and 42 Scopus. She has supervised 9 PhD scholars, 4 granted patents, and has received research grants as PI and co-PI. Her honors include ISEB Fellow 2025, National Women Scientist Award 2025, Women's Achiever Award 2023, Wari Fellowship USA, and multiple national recognitions. Madam, I welcome you in this session, and you are expected to complete your presentation within 20 minutes. Welcome, madam.
Thank you, sir, and thank you for informing me about the time. Although I think my time of the presentation from 3:30, and I am also waiting from the last 30 minutes. So I will try my best to complete within the time limit.
Yes, ma'am. Thank you so much for your cooperation and understanding. You know, the conferences generally get delayed by 15 minutes.
Just get delayed, but you have to inform me also because I have some other assignments also. I am a little bit bound that also.
Okay, ma'am. Please.
Okay. So thank you for giving me this opportunity to share my views on this current topic. And it's really good to hear about the Dr. Richi Mahajan from Department of Biotechnology and his views on the why we are working and what are the different challenges and what are the needs should be done to make the technologies from lab to land approach. It is really needed. So is it my screen is visible?
Yes, ma'am, your screen is visible now.
Okay, thank you.
Also, ma'am, if you can turn on your camera.
I'm doing this, but sometime network issue is here. So maybe it become difficult to, maybe that voice will be break. Okay.
Thank you. Yeah. You're requested to kindly keep your slides in presentation mode so that it is full.
Is it okay?
And you can press F5 from your laptop. F5.
This is not working. Sometimes it worked and sometimes not worked. Yes. So So my topic for the presentation is bio-innovations for circular economy. And what are the different emerging trends and strategic action should be needed to make it more on the part of commercialization. In this, my slides, I have worked on these agendas: a brief introduction to circular economy, then circular economy core principles, what is circular bioeconomy, why it is important, why the and what is bio-innovations, key drivers for bio-innovations, emerging trends in bio-innovation, alignment with SDGs, which is the demand for society nowadays, what are the different strategic actions, global momentum towards policy and investment, in India, how it's emerging on the part of bioeconomy or bio-E3 policy. Then I have tried to conclude with different case studies which are on the part of working on the circular economy. So as we all know that, as Dr. Garima and Dr. Santosh also mentioned that we have to move from information to innovations. On that part, circular economy is one of the things we have to work on. It is an economic model that promotes circular production and consumption cycle, and it should be how to make, how to use, how to collect, how to transform, and how to reuse. So these are the basic kind of things which should be known on the part of circular economy. Its core principle is design out waste and pollution from the system. Then keep products and material in use at highest value. Then regenerate natural systems through biological cycles. These are the main part. Then we circular economy convert to the bioeconomy, when the biological things has been used with the proper strategic actions. Then shift from linear take, make, dispose to circular loops. So circular bioeconomy is integration of circular economy principles with biological resource utilization. It utilizes renewable biological resources sustainability. It reduces reliance on fossil fuels, decrease greenhouse gas emissions, and promotes waste to value transformation. Crisis of why this take, make, waste model has now on its maximum carrying capacity for any ecosystem. So resource depletion is also the problem. Annual global extraction of materials has grown from 30.9 billion tons in 1970 to 95 billion tons in 2020 and reaching 106.6 billion tons in 2024. It is an annual average growth rate of 2.3%. And climate pressure is also there, because we are talking about there is increase in temperature of 1.5 degrees Celsius. So the cause of the melting of ice glaciers and ultimately the side-by areas of the coastal regions, it may be merged into the water. So circularity gap, despite global efforts, the world is only 6.9% circular in 2025, a decline from 7.2% in 2023 due to soaring consumption. Then there is a term is bio-innovation, although innovation we all are known to us. And what is bio-innovation? It is a biological innovation, refers to the development and application of novel ideas, methods, technologies, or processes in the field of biology, where solving biological challenges, advancing scientific knowledge with scientific knowledge is the important thing, and addressing practical problems. Because when we talk about the any advancement in scientific knowledge or scientific technology, then the practical problems are always differ from land, if we talk about lab to land approach. Bio-innovation include biotechnological advancements like CRISPR gene editing, as Dr. Ritchie well mentioned in the previous presentation. Then CRISPR gene editing, biomimicry, bio-based materials are there, sustainable solutions such as biofuels or bioremediation. So in this bioeconomic context, renewable biomass used for products, services, and energy with the reduced use of fossil fuels, dependence, and supporting circularity is important thing. Environmental imperatives means we have to directly or indirectly relate all the things or all the commercial applications we are talking about belongs to environmental imperatives, then it should not contribute to the climate change, biodiversity conservation, resource depletion concerns. Then technological advances should be known to us: rapid, cost-effective gene sequencing, AI, data science, synthetic biology are accelerating biological research and applications. Market demand and consumer awareness is required. Growing consumer preference for sustainable bio-based products or eco-friendly products, or the products which should be eco-labels. Agriculture and industrial efficiency, demand for higher food security through biotechnology, particular to the drought-tolerant crops, fertile soil with the help of with the minimal use of chemical fertilizers, biofortification, and improved industrial processes like bioconversion and fermentation. So circular economy plus bio-innovation is the game changer in the present scenario. It is a symbiotic partnership where waste is used as a feedstock. Because biological systems do not recognize waste, bio-innovations turn agricultural residues and food waste into high-value chemicals and textiles. Decoupling growth enables economic expansion without relying on finite virgin minerals. Value creation and cascading use. Again, the use of biomass first for high-value products, just like pharmaceuticals. Then the materials, the side-by products can be used for the other purposes, for the other, make it into a other value-added product. And finally, it can be converted into energy also. So carbon management, capturing CO2 and using microbes to convert into protein or fuels. An outcome, a system does not do just do less harm, but actively regenerate the ecosystem it depends on. So this is game changer, should be promoted by all the policy part and as well as the industrial and institutional levels. What are the different emerging trends in bio-innovations? Waste valorization and biorefinery approaches means use of agriculture waste or the forest waste or the industrial organic waste, and it should be converted into a value-added product. Then microbial biotechnology and enzymes are there under the part of the bio-innovations. Biofabrication and next generation biomaterials. As the whole world is working on the part of AI, artificial intelligence. So digital integration in biotechnology is one of the important thing, and it should be included with the bio-innovations. Then microalgae and carbon capture is there. If we talk about number one, waste valorization and biorefinery approach, there are the lignocellulosic biorefineries, which involve advanced conversion of biomass into diverse product streams. And what are the different bioproducts can be formed at biofuels, and energy is there. On the part of biofuels, bioethanol, bio-oil and syngas, pellets and briquettes, and biogas. Then biochemicals and platform chemicals are there, and biomaterials and function materials. Different nanocellulosic materials are there, using to make the more efficient technology from industrial purposes. Then activated carbon is there. Resins, binders, carbon fibers, phenolic compounds. These are all the bioprocesses. If we go through them with the efficient part, with the use of the efficient materials, then it can be a fruitful solution. Then microbial biotechnology and enzymes, breaking down the unrecyclable. The limitation is mechanical recycling degrades material quality over time. But with the bio-innovation, engineered enzymes to depolymerize complex plastics into base monomers, and the result is infinite recycling loops that produce virgin quality materials with significantly lower energy inputs. Third emerging trend is biofabrication and next-gen biomaterials. It growing our materials means use of the materials has been increasing as per the demand, and particularly instead of biochemical based materials, we should work on the bio-based materials, utilizing living cells and precision fermentation to grow materials rather than extract them. Particular applications are mycelium-based packaging and construction materials, brewed proteins for textiles, and PHA, that is the polyhydroxyalkanoates, marine degradable bioplastics. AI and digital integration in biotech, the digital bioconvergence with the AI protein design, then IoT sensors tracking biomass availability and waste streams in real time is required. Digital product passports, blockchain-enabled tracking of products, biogenic content, and circulatory matrices are required. If we talk about microalgae and carbon capture, so nutrient removal coupled with production of high-value lipids and pigments, and it is a very new topic on the part of that how we can use algae for the carbon capturing. Although it is this process or mechanism is not new. This is the conventional process or mechanism which the algae is following to convert to capture the carbon. But now the technologies are available from where this can be scientifically required in the present scenario. Then integrated CO2 capture, direct fixation of industrial emissions combined with biofuel production via in situ transesterification. So it creates a double benefit for the climate action. Then AI-enabled monitoring, advanced systems for algal growth with the, if we talk about the controlled conditions here, then this AI-enabled monitoring with the photobioreactors optimizes yields and prevent culture crashes. SDGs align with the bio-innovations are affordable and clean energy, decent work and economic SDG 7, 8, 9, 11, 12, and 13. These are the major SDGs which are connecting with bio-innovations to global goals. If we talk about the national strategies, then circular economy and bioeconomy roadmaps across ministries create an unified frameworks that prevent policy fragmentation and support integrated development goals. Life cycle assessment mandates are required, digital product passports, DPPs, certified composability and biodegradability. So on the part of policy and regulatory frameworks, these standards and matrices need to be completed. Then strategic action 2, that investment in infrastructure scaling, moving innovations from lab scale to commercial scale production. This is one of the major challenge because if we talk about the lab scale, where we are working mainly on the control conditions. But on the part of commercial scale, then we have multiple variables which may or may not be directly or indirectly impact the processes or mechanisms. So solution, targeted funding for regional decentralized biorefineries, financing, fostering public-private partnerships, and mobilizing green capital to de-risk early-stage biomanufacturing facilities. Third strategic action should be on the part of open innovation and cross-sector collaborations. Breaking down the industry barriers, interdisciplinary actions are required on the part of uniting agriculture, biotechnology, waste management, and traditional manufacturing. Knowledge sharing is required. Sometimes indigenous knowledges are available or indigenous techniques or technologies are available, which should be shared with the regional as well as national level to make it more compatible for the society. Promoting open source databases for biocatalyst discovery and material flows and inclusive growth, engaging local communities, farmers, and indigenous populations in managing bioresource sustainability. If we talk about global momentum and policy and investment, then European Union, United States, China, these are the some major investors on the part of policy framework. At the national level, we have Bio-E3 policy, as mentioned by my previous speaker also, that biotechnology for bioeconomy, environment, and employment. So core framework for circular bioeconomy is mainly object with the manufacturing program will steer India on the path of accelerated green growth by promoting circular bioeconomy, transformative shift from consumptive manufacturing to regenerative principles using microbial, plant, animal, and human cells for commercial products. Net zero alignment as the India's mission to net zero by 2070 of the CO2 goal through the bio-based products with minimal carbon footprints. So target double economy by 2030. Key focus areas: research, innovation for climate change and decarbonization, stronger domestic scaling up, pilot and pre-commercial biomanufacturing capacity, high performance processes, leveraging living systems, promotion of bio-based products in food, health, agribiology, marine and space. So bio enablers are there, the implementation infrastructure. Biofoundries and biomanufacturing hubs are required. Bio AI hubs are required. Bio E3 strain research centers for fostering high performance manufacturing. Just like NCCCS Pune is there, provides access to high performance indigenous microbial strains with freedom to operate status, facilitating efficient high performance biomanufacturing processes. Some case studies, just like Praj Industry is there, who is working on 2G ethanol from agricultural residues. Praj Industry, it located in Maharashtra. It conversion of rice straw and crop residue into second generation bioethanol. Lignocellulosic biorefinery model is there, and advanced enzymatic hydrolysis and fermentation technology. C6 energy seaweed based bioproducts by this company located from in Bangalore. Large scale cultivation of tropical seaweed, production of biostimulants, bio-based chemicals, and functional food ingredients. Then one company is Fool.co, who is working on floral waste and convert it into biomaterials, upcycling temple floral waste, and produces biodegradable packaging, natural incense, mycelium-based leather alternative. Then Bio-E3 Strain Resource Center, High Performance Biomanufacturing. It is under the DBT, and research area focuses on stem cell biology, immunology, microbiology, and genomics. And infrastructure, a national repository for animal cell cultures, an experimental facility, and specialized labs. Biofuel Junction in Maharashtra, focusing on aggregating agricultural and food waste to produce and supply quality biomass briquettes and pellets to large industrial clients, filling a gap in supply chain reliability. So if we talk about the final thought of this presentation, that transition to a biocircular economy represents a transformative shift from linear extractive system to regenerative and restorative models of growth. By integrating circular economy principle with cutting-edge bio-innovations, such as microbial biotechnology, biorefineries, AI-driven biomanufacturing, and carbon capture waste can be converted into wealth, decouple economic growth from resource depletion and leading to ecosystem restoration. In this part, I also wish to add in my lab, our lab students working on the part of waste minimization with the help of how we can convert organic waste content into the bioplastics and biopellets. We are working in the lab on that part also. A strategic biopolicy framework is required. Investment in scalable infrastructure and cross-sector collaborations are critical to accelerating this transition. So it should be needed a proper attention. With a strong global momentum and initiatives like India's Bio-E3 policy, the path toward a sustainable, resilient, and low-carbon future is becoming clearer. So this is all from my side. Thank you.
Okay, thank you, Dr. Nisha. And you are very well explained different concepts of bio, circular economy, including different case studies that have observed. One of the
Sir, your voice is not clear to me. Maybe.
hello, ma'am.
Yes.
Formative lecture, and you have explained all the concepts of bio-innovation, circular economy, and sustainable development, including different case studies. Hopefully, the audience will enrich this knowledge, and definitely they will implement all these knowledge in their upcoming research. Thank you so much for delivering such a wonderful talk. Madam, just I want to know what is the major challenges that we are facing when we are converting any technology at industrial scale.
The major challenge is one is the funding, because when we talk about the lab scale, we work on the small or under the control conditions. So funding is one of the important thing. If that is given by the funders, or the industrial collaborations if there, or the patent type of technologies are there, that will promote the lab to land approach.
Audience, if you have any questions, you can ask.
Any questions from the audience?
Okay, thank you so much, madam.
Thank you, sir.
Thank you
And
so
thank you for again giving me this opportunity.
Thank you so much, ma'am. We really appreciate it.
Thank you.
Can we have a huge round of applause for her? So before moving on to the next session, I would like to make a small announcement that Dr. Richi Mahajan will be leaving for his commitments. So I request session chair, sir, please felicitate Dr. Richi Mahajan along with Dr. Nidhi Chaudhary ma'am, Vivek sir, Amit sir, please do the honors. Please give him a huge round of applause for a wonderful, wonderful talk. Thank you very much. So we'll now continue with the session.
To continue further this session, I invite Dr. Nidhi Chaudhary, madam, to deliver his talk. Dr. Nidhi Chaudhary is a professor at the MIT Institute of Biotechnology, MIT University, Greater Noida. She has 29 years of teaching and 26 years of research experience and 14 years of administrative experience. She served as head Center for Biotechnology and Biochemical Engineering for 10 years and currently served as head Innovation and Entrepreneurship Cell, Life Sciences Domains, and is a member of the Institution Innovation Council, Ministry of Education, Government of India. Her research focuses on industrial and therapeutic enzymes, bioprocess technology, and well-form-based strategies. She has made notable contribution to translational research, including technology transfer of a novel protease enzyme to an Indian biotech startup, with several technologies in the commercialization pipeline. Dr. Chaudhary holds 18 patents, out of these 2 granted, has authored 78 research papers and 11 book chapters, and has supervised 8 PhD, 1 MPhil scholar, and over 240 UG and PG students. She is a recipient of 21 national and international awards. She was conferred the SPSR Outstanding Women Academic Leader Award 2025 and is a fellow of the Indian Society of Agriculture Biochemist. On the behalf of this session, I welcome you, madam. and please deliver your talk. And we are expecting that you will complete your talk within 20 minutes. Welcome, madam.
Very good evening, everyone. Well, yeah, this is late hour. I know, I understand that. But please bear with me. Lot of the topics have already been aligned and told by the two speakers before. So I'll be skipping through them and we'll be moving fast. Well, out of the 9 planets in our solar system, what makes Earth unique? Can anyone tell why Earth is unique? Earth is unique due to 2 things. One, that is water, presence of water. And second, because of water, the elements present on the Earth and in the environment on the Earth, they interacted and made living system, right? That is archaebacteria, then further finally evolved human being. So there are 2 things which make Earth unique, that is water followed by human beings. And that is why we are standing here holding this conference and many of such conferences so that the Earth, the human beings remain sustainable. We are precious. Remember, this Earth is precious. Human beings are precious. So we are talking about sustainability of Earth, the planet, the human beings who are residing on it. So throughout whatever we are doing, we have to keep a focus. Whatever act, even a minute act of ours, is affecting the sustainability level of Earth. We have enough destroyed it, but now we have to correct it. We have time. We have to take corrective measures and align in a manner that it remains sustainable. So with this starting note, let us move through the journey how we have reached to this state by doing what we call industrial revolutions. And after doing industrial revolutions, we have reached to a state that we are completing this circle and trying to reach to a state to save ourselves from ourselves, right? So coming to industry revolution, significance of biomanufacturing for humankind. But first, of course, we have to retain and sustain the humankind. So let us travel through the industrial revolution. Before industrial revolution, when civilization started, when humans became civilized, they started with agriculture. First, they were just behaving like animals, just eating raw, then started killing animals, innovated some arrows, bows, etc. Then came the biggest innovation that human beings did, that was of wheel. Wheel is the biggest innovation that human beings did. And believe you me, this is the one which can beat none. It is the wheel which has turned everything around human beings. Then came series later on, but it was the wheel which changed the pace of revolution. So it went on and on. Then humans started with agriculture and handicraft. Barter system started. So economy was all just barter system. Then came this, made some coin-like structure, and they started giving, sharing them and work. So actual industrial revolution started in 1760 when it was United Kingdom, and followed by United States, France, and Germany, who worked at a faster pace. And from agriculture and handicraft, they started moving to industry and machinery. So now came the role of machines. And the term, it came from a French word, la révolution industrielle, and further, rest is history. We get to know what all levels we have reached. So now we are in the 5th generation. We have entered into the 5th generation of industrial revolution. So first industrial revolution, that was steam engine. Then your mechanization power, that was in 1800, very big jump in life of human being. Life, the pace of human being lives started at a very high, with acceleration, and large number of huge load could be taken from one place to the other on a small, containers running on rails. So that increased the pace, movement increased, and civilization started developing from one part of the country or the countries to the other. Then came two, where electric power played the major role and mass production started in Industrial Revolution 3, that is in 2000. Now the pace started from 1760, and now it increased at such a pace that within 300 years, we have reached to a stage that now we need to put brakes. We are at a stage that brakes need to be found, and in what manner we have to put the brake and which brake is to be put, that we need to sort out. So then started with computers in 2000, that is in Industrial Revolution 3. In 4 started with your machine data, IoT, artificial intelligence, that rules. And now we are into Industrial Revolution 5, where we have to think in a sensible manner. We have to take help of robots. We need to take help of AI and computer system, but in sensible manner. That is, we need to work out with cobots, collaborative robots, and have human intervention. So that is what is required. That is sensible use of robotic systems, sensible use of computer system, and this artificial intelligence. That's the buzzword now. So these are just few examples which I'll move on fast. That is in 18th century, Industry 1.0, steam engines in case of your machinery, but in biological processes, we were into fermentation, into brewing, etc. In 2, that was in early 20th, there was electrification, and we were using the microbial processes for production of different products. In 3, that is in late 20th century, there was computer system. And in case of biological systems, we had recombinant DNA usage and production of insulin by using that. In 4th, we used IoT, AI, big data, and that increases the efficiency. In very short period of time, we got lot of data. We could use it and do number of things at a short space of time. If you talk of biological system, not many science developed like metabolomics, genomics, transcriptomics, etc. with the advent of computer systems. So our whole data can be worked out in a single go and we can get in silico analysis done. Now come Industry 5.0, human-centric, where we are at present. This is, my dear, where we need to think in a very sensible manner. I'm repeating it because this is a highly sensible zone, sensitive and sensible zone. Because of such very thin line, there is a very thin line. If it is being crossed, then everything will go to zero. So human-centric is required. We can take help of all these systems, but for getting collation, analysis of data, but finally give human touch to it, then only it will be of benefit to us. If we compare all these 5 generations of industrial revolution, this is a comprehensive view. The most important part, rest we can all see. Most important where I want your attention is the 5th generation where we are now, which need clean energy, human-centered technology, human-machine collaboration, AI plus ethics. We need ethics here. We need cobots, not robots. Collaborative robots, human intervention plus robots with sustainable technology. We need personalized and sustainable production of different products, human-centric innovations. Sustainable and inclusive economy will only be reached by this process. And so finally, we'll be reaching to the overall well-being of human beings, all livelihood, plants, planet, and the whole solar system. And of course, different countries have been working in these industrial revolutions time to time. Now, not many countries are taking initiatives, and out of them, India is coming up at very fast pace. Now, coming to biomanufacturing, now when we talk of human sustainability, one thing is the day-to-day products which we use, which we consume, which we use. So when we see our household things, whatever we consume throughout the day, whatever we wear or use throughout, that all need to be manufactured. And it can be manufactured either by chemical process or by biological process. So now, for sustainability, we need biological processes, that is intervention of microbial systems, so that it gives us a clean technology, which is environment friendly. So biomanufacturing refers to use of engineered microbial plant and animal, including human cells, for increasing precision and control to produce commercially important products, that is fuels, most important, your biofertilizers, medicines, enzymes, food, etc. on large scale. So that is the target. All government schemes you are seeing, they are majorly targeting towards biomanufacturing. So these are versatile processes and they have the potential to create the bio-based products, allowing efficient utilization of resources in a scalable, cost-effective, sustainable, and environment-friendly approach. Each word needs to fit in the process. Then and only then we'll get to know. Dr. Mahajan mentioned biomanufacturing. It seems easy, but it is difficult, but it is challenging. That is why we are talking about it so much. Once it will become easy to do, then we won't discuss. It will become a routine process. So why it is important for humankind? It can address climate change and resource depletion, support affordable healthcare and food security, reduce carbon footprint of industrial processes. For environmental sustainability is of immense importance. And of course, the cleaner production. This slide has already been shown. What is the India's bioeconomy? At present, we are at around $160 billion, and the target by 2030 is to reach to $300 billion. So it is of immense importance. And what is biomanufacturing? It is composed of two steps. One is called upstreaming and other one is called downstreaming. Upstreaming means production of any desired product, and downstreaming is to get the desired product out of the whole lot, which is actually a mix of everything undesired or byproduct, which we do not want at that point of time. So this is a simple continuous biomanufacturing unit, which you all see. Now, applications of biomanufacturing is in almost all the areas, touching all areas of our life. Pharma, biopharma, food beverage, agriculture, enzymes, biochemicals, and all miscellaneous like bioplastics, biofabrications, bioremediations, now cosmetics, cosmeceuticals, everywhere. So biofermenter, it is a typical one. We have a system into which we put the medium plus inoculum, that is the microbial system, which is producing the product which you are desiring, and further when it has reached to the desired concentration, then we take it out and downstream. So that is upstream, downstream, that is what we need. And of course, there need to be quality check in between, and there have to be some quality safety steps that need to be followed. So upstream, as mentioned, you have to put the suitable conditions in various fermenter. Fermenter type also differs according to the product which you are interested in and the microorganism which you are using, or the cells, whatever you are using. Downstream, when we extract, and with each step, we increase the purity level till we reach to the level that we intend to, or at the level at which we want to sell it into the market. That is called downstreaming. Only after downstreaming do we get the value for the product. So quality check is most important. Is it meeting the standard of the global market? Whatever you have prepared, that need to pass through all the benchmarks, and then only you can say that the product formed after biomanufacturing is worth taking to the market. Now we are getting the units in or the bioreactors which are automated. We have robots and AI which are optimizing the bioreactors, but they are costly. But as this will go at larger scale, the cost will go down, and that is the target what we are now moving towards. Now, challenges. There are many challenges which we face in biomanufacturing. Scaling up, as Dr. Mahajan mentioned, what we get in flask, exactly replica, we do not get at large scale. So at every volume increase, the conditions differ. So every time we need to optimize. Once we completely close the chamber of a big bioreactor, we do not know what is happening inside. So it gives totally different results. So scaling up strategies need to be refined, and they need to be optimized every time. Production need to be monitored. Cost analysis is to be done. Budgeting, resource management, testing, regulatory compliance, very important. Every product has to pass through all the government policies. Risk assessment. Is the product safe? Everything is to be tested, and only after all the testings does the product goes into the market. These are just the examples. These things I have already covered. So there are no level of acceptance at present, but the acceptance will increase with time. Every new thing comes with challenges, but with time we sort it out. There are technical limitations, regulatory aspects are there, cost factor is there, but solutions are there. That is, when there will be increased demand, obviously the cost will go down. Manufacturers need to make more and more of innovative bioreactors. We need to collaborate so that any good bioreactor, automated good bioreactor, does not remain empty for some time. We can use their bioreactor and manufacture the product. So instruments should not lie vacant so that the equipments can be used to optimum level. That way, we can increase the biomanufacturing level. Benefits are energy use reduction, supply chain optimization, increased innovation, and sustainability, which are the main targets. So why it matters for humankind? Obviously, it matters. All the products, as I mentioned, this is just repetition. Every product can be made at larger scale, at low cost. Maybe biofertilizer, biopesticide, any nutraceutical, then pharmaceutical, etc. Everything can be produced at lower cost in less time period. So that has immense role in all the sectors. So if we go sector wise, I'll move fast. I think there is paucity of time, sir. So not many things. It is very important in agriculture and food. That is the main sector because human beings first need food. So that is the first sector that requires biomanufacturing. First, and climate, of course, climate and food both are very important. Carbon footprint need to be reduced. These points are already covered. Environment, we need to address the problem of environment. Then biofuel, then in healthcare, vaccine, therapeutic proteins, monoclonal antibodies, gene therapy, all these. And we already have seen biomanufacturing as a game changer during COVID era. How vaccine was prepared in very short period of time when the companies worked together and made large quantity of the vaccine, which sorted out the problem. So how it impacts the economic and social factors? It creates jobs. It makes startup ecosystem, bioeconomic growth. So it meets the India's vision of sustainable development, innovation leadership, and that lead to self-reliant India, that is Atmanirbhar Bharat. So we can say that biology is driving manufacturing into the future. So now it is the time of biology which will drive it. These are the government initiatives which have already been discussed. So I'll move on very fast, which are actually targeting exactly towards biomanufacturing, Bio-E3 policy, having biotechnology for economy, environment, and employment. So these all you can see at PPT site. These 6 are the thematic areas of biomanufacturing. So whenever, for students especially, whenever you target your area of research work, first see the government policy and the burning issues which are floated on government portal, ministry portal. Every ministry floats the areas, thematic areas which they want solution in, and according to that they float the scheme. So find out that area which matches with that and then further start working into it. So these are the supporting initiatives which government has started. Startup India in 2016, Make in India, then production linked incentive schemes for biopharma, skill development, and regional biotech clusters. Along with that, we have other schemes for having incubation. Challenges: how to scale up from lab to industry, investment and skilled workforce need, and strong industry academia collaboration. So how to go ahead? The problem is that all are working very nicely. Students are doing very nice. Industry is doing good. Academia is doing good, but there is a gap. So we need to overlap, sync all the three and sync it in a manner that government need to pitch in with some policy that they have to work together and give some funding or some policy that industry need to work hand in hand with academia to work out for a solution. So we need to adopt net zero energy as well as carbon footprint technology, global public-private partnerships, and effective collaboration. Now I'll show glimpses of work which my lab is doing towards the initial phase that may lead to biomanufacturing. In fact, one has gone for technology transfer. So in my lab, we have been working on different technical, industrial, and therapeutic enzymes, and we have found few novel enzymes, multifunctional novel enzymes, thermostable enzymes. Here, this is just the zest of enzymes and their properties, which are of industrial usage. This is the technology that we transferred in 2023 to a company for biomanufacturing, but only for a specific application, food and beverage. Other technologies are at different levels and ready for either technology transfer or for biomanufacturing. So we can conclude that biomanufacturing is the backbone of the next industrial revolution, and India is strategically positioned to lead globally, and sustainable growth will be beneficial for humankind, and that is in alignment with nation's mandate to reach and make India Atmanirbhar Bharat, Viksit Bharat by 2047. So that is the global vision and mission that we have to work together towards human sustainability. So human sustainability means earth sustainability. So overall, we have to work towards that mission. Then and only then we all will be happy go together, and we'll have nice.
Thank you, Dr. Nidhi, for delivering such a wonderful talk. And you have covered the different industrial revolution significance and bioinformatics for your biomanufacturing and different aspects of application of bioinformatics for humankind. Thank you so much for delivering a wonderful talk. Thank you, madam. And our next speaker, he is present online. Now I would like to invite Dr. Binod Kumar. Dr. Binod Kumar is presently working as a senior scientist, fermentation and microbial biotechnology division at the CSIR Indian Institute of Integrative Medicine, Jammu. He obtained doctorate from the University of Delhi, for which he had obtained a fellowship from MNRE, New Delhi. Before that, he worked at CIAB Mohali as technical officer, microbial technology, research associate and young scientist under the DST SERV, Government of India. He was awarded FPFL Visiting Fellowship in 2016 and worked there during September to October 2016. His current research interest is production of bacterial cellulose and preparation of composite of bacterial cellulose, xylitol, and industrial important enzymes from unutilized bioresources and processing residues. His main research interests are in the area of industrial microbiology and biotechnology. He has published 76 papers and 5 patents filed and 3 grants. He is a member of Biotech Research Society of India, Society of Biological Chemists India, and Center for Energy and Environment Sustainability India. He has visited to Korea, China, and Switzerland. Dr. Binod is serving as an editorial board member of Journal of Innovation in Applied Research. On the behalf of conference, I welcome you, sir. Please deliver your talk, and we expect that you can complete your talk within 20 minutes.
ठीक है, थैंक यू सो मच सर. फर्स्ट ऑफ थिंग्स, ये सॉरी फॉर द मैं टाइम पे अटेंड नहीं कर पाया और इन फिजिकल अटेंड नहीं कर पाया. क्या मेरी स्लाइड विजुअल है सर? सर मेरी स्लाइड विजुअल है?
यस सर, इट इज विजुअल सर.
और सर और आवाज भी आ रही है, ऑडिबल?
नहीं आ रही सर, आपकी आवाज आ रही है.
Yes, sir. You are audible. You are audible.
Yes, sir. So first of thanks, good afternoon, sir. Myself, Dr. Binod Kumar, thank you, sir, for providing me this opportunity. And today's my topic: enhanced xylitol production using the non-detoxified xylose-rich and partially detoxified and pre-hydrolyzed spent biomass and newly isolated from the bacteria yeast. And so, sir, I am working from the CSR laboratory, and all of you know that CSR laboratory in all over India is 37 laboratory and 39 outreach center in our country. and I am working on the CSR Triple M Jammu and the Jammu region, J&K region. This is the focus on the biological science, and this our institute in focus on the medicinal chemistry and pharmacokinetics, formulations development, and biotechnology, plant microbial secondary metabolites. And I am working on the fermentation based technologies for the industrial products, and we are also GLP and CGMP facilities available, NABL and QC, QA, direct food essentials is all, and pharmacology cancers and inflammation. These all the facilities available in our lab, and different group is working on this rope. So our division is mainly working on the product bioprocessing. It may, matlab, we are working on the basically develop the process for the production of any bioactive molecules up to the pilot scale. And this, what is the between the gap between the industrial scale? So how to rectify this process and should be cost effective manner and different way. So we are developed on the process and for the industrial perspective. Second thing, spent. Why are choosing the spent biomass? Spent biomass is also a lignocellulosic biomass, which is a huge amount is generate in our country as well as in world. If 4,000 is 400 plant is already is available and cultivated for the aromatic plants, and spent biomass is generated after the extraction of oil. Is a huge amount of the biomass, and right now used as a burning. And extraction of the is approximately India is being the worldwide leader is citronella is a mainly aromatic biomass is lemongrass is a huge amount of main jo leader has spent biomass means the lemongrass. So 6 million ton is per annum spent aromatic rice is generated in our country. So sir, ये कि spent aromatic spent biomass is containing is a hemicellulose content, cellulose content, and lignin content. Depend on the their quantities also depend on the type of the aromatic biomass. So besides, and this is the also we are choose the lemongrass, and they contain approximately 35 to 40% of the cellulose content, and 25 to 30% of the contain hemicellulose, and remaining the lignin production. So xylitol is a pentose sugar alcohol, and this is the extract production from the xylose. Xylose is the pentose sugar, and this is the available in the main focus of the hemicellulose. So we are focused on the hemicellulose content for the extraction of the xylose content, and then further the fermentation to the xylitol productions. So this is our research plan, sir. Key aromatic plant take the cut the aromatic collect the aromatic crop, then distillation, and one product is essential oil, and after the spent aromatic biomass is based is generated, and the pre-treatment process, then hydrolysis, and this fermentable sugar is the fermentation, and then the recovery of the best spent biomass. And other lignocellulosic biomass is benefit in a case of the spent biomass, because this is the hydrothermal, because in normally is a rice straw and wheat straw is contain the lignocellulosic biomass, but they're not treated. So it's already this process is complete in during the distillation. This is a mild hydrolysis already is there. So some detoxification is maximum inhibitors is compound is already removed during the distillation process. So this is our good opportunity ki we can some mild process is there, and then extraction of the sugars. This is the up now is the xylitol. What is the xylitol? Xylitol is the pentose sugar alcohol, and this is the produced from the xylose content, sir. This is a 5 carbon sugar, and this is a very approximately low calorie compared to the sucrose. And there is the molecular weight is a 152, and xylose molecular weight approximately 52. And this is the reduced. This is used as a dental caries also. And there is chemical feature is a protein and melting point 92 to 98 degrees centigrade, and global market. So this is the approximately high value product. You can say high value product is also, and it is a very demand product, not a high value, because this already is available in market 3 to 400 rupees kg. There is a huge amount of market is right now, and what low calorie sugar 1.4 to 7 is approximately in 2013, approximately 4.7% depending on the forecast shows. In the chewing gum industry is a major consume this low calorie sugar alcohol in the right now, and 53% approximately 50% of the xylitol is consumed in the chewing gum industries. This is the market value. Right now is China is the major source for the production of xylitol is from the corn cob, because corn cob and dew point is USA is also European countries also produce the xylitol from the birch tree. Corn cob is contained approximately 35 to 40% of the hemicellulose content, or birch tree is contained approximately 50 to 60% of the hemicellulose content. Due to this reason, this selected the crop. Now this is the xylitol market. European countries is produce sale approximately 860 rupees per kg, and the Chinese market is produced 400 to 500 rupees per kg. And 50% of the xylitol is produced in the China by the chemically routes and European countries. This is the application of the xylitol, and low calorie alcohols, toothpaste, and mainly diabetic patients can use this use in the right now pharmaceutical syrup or mouthwash or chewing gum and so on. So right now is currently is the xylitol is produced in a synthetic route, is acid hydrolysis and rayon nickel, and this is especially is required the some pressure and temperature is conditions, specific conditions required for the production of synthesis of the xylitol. But problem is that after the some time is that some rayon nickel is this the carcinogenic and highly required the purified substrate. In chemical reaction is required the highly purified of the substrate. Due to this reason, is agri residues is very difficult to convert the xylose and then ferment to xylitol. But in the case of the fermentation, we can easily fermentation of the xylose to the xylitol by using the dehydrogenase enzyme. And in the case of the fermentation, we cannot require a very specific and highly purified substrate for the xylose. Now this is the extraction of the xylose from the no. Sorry, sir. Some.
Hello, sir. Your slide is not visible.
Wait, sir. थोड़ा सा internet issue. Hello.
Yes, sir. Yes, sir.
सर आवाज आ रही? हेलो
Hello.
सर आवाज आ रही?
Visual, you can continue.
Sorry, sir.
Yes, sir. Now it is visible.
तो सर हमने जो एसिड हाइड्रोलिसिस किया उसमें से हमने करीब करीब 0.9%, 1%, 1.5% ऑफ एक्सट्रैक्शन ऑफ जाइलोस फ्रॉम द biomass is successfully using the nitric acid and 0.5% concentration, and but in the some detoxify inhibitors compound is also generated in the acid hydrolysis. So these molecules is interfere in the recovery of the xylitol, because during the acid hydrolysis some inhibitors compound is also produced, and this is the very difficult to recovery interfere in the downstream processing of the xylitol crystallization. So second one more challenge, sir, is that the acid hydrolysis detoxification of the process already is contain the your process should be cost effective manner, because of very limited, because already 400 to 500 rupees of xylitol is produced, market is supplying. So if we are adding the number of steps, their process cost is also increased. So we are limited to how to remove this remove the inhibitors. So we are using the membrane filtration, and successfully approximately 73% of the 72% of the xylose from the spent biomass as the highly specific, highly and also salt, lignin, and phenolic content removed from this membrane filtration. But drawback of the membrane filtration, sir, we can if we are using in batch process, because batch process we are using the membrane filtration in batch process, so very loss is there. अगर हम कोई प्रोसेस को कॉन्टिन्यूस प्रोसेस में डिटॉक्सिफिकेशन करेंगे, अगर मेम्ब्रेन फिल्ट्रेशन यूज करेंगे, तो इट इज द बेनिफिट कंपेयर टू द अगर हम इसको कंटिन्यूस प्रोसेस करेंगे तो मेम्ब्रेन फिल्ट्रेशन सफिशिएंट फॉर द एंड इजी रिकवरी फॉर द xylose concentration and effectively remove of the minor inhibitors. But in batch case, so we are more loss of the product. You can see 72% of the xylose is only the required and approximately 28% of the loss in the batch processor. So after some time, sir, some salt, because the acidic hydrolysis is pH is contained the 2 से 3, तो जब हम neutralize करते हैं, तो there is the process increase and some salt is there. So this salt cannot be easily removed by the membrane filtration. So we are using the ion exchange process. Ion exchange process is also cost effectively because sir, we can use the resin is many times based on the your handling and based on the process. So approximately 68% of the salt is removed and 92% of the xylose is also recovered from this processor. तो जब हमने इसको प्रोसेस किया जेल्टोल को फ्लास्क पे लगाया तो सर हमने देखा कि अप्रोक्सीमेटली प्योर जाइलोस था 78% ऑफ जेल्टोल इज प्रोड्यूस एंड आफ्टर द डायरेक्ट हाइड्रोलिसिस किया तो उसमें हमें कोई यील्ड नहीं आई नो माइक्रोबियल ग्रोथ एंड पार्शली प्यूरिफाइड एंड लेस डिटॉक्सिफाइड प्रोसेस इज सक्सेसफुली 45% of the xylitol is produced. Now this is the 5% of xylose contained take in the fermenter level and xylitol is successfully 34.6 gram, approximately 67% of conversion, 63% conversion and productivity is there 0.6 gram, मतलब 1 gram of xylose से हमें 0.67 gram of the xylose मिला जो इस fermentation process से. अब नाउ इज द सेकंड चैलेंज इज द कि जाइटॉल जो रिकवर किया था हमने फिर फर्मेंटेशन के पहले जो अपस्ट्रीम प्रोसेस में जो हमने प्रोसेस अप्लाई किया तो दिस प्रोसेस सुटेबल आफ्टर द फर्मेंटेशन इज गुड और नॉट तो वी आर यूजिंग द लॉट ऑफ द लॉस 52% of because the xylitol property is different and xylose property is different so this is the very difficult to extract the xylitol from the fermentation after the fermentation same process को apply करना is very difficult. So we are using the another xylitol refining. This process already right now ongoing in the case of the sugar. Table sugar will purify carbonation and ion exchange process. In this process, calcium carbon dioxide and carbon dioxide at the reaction and can take the calcium carbonate and this calcium carbonate is absorb the color gum amino acid impurities and precipitate in the form of the calcium carbonate and easily remove and action ion exchange chromatography one more step is also can be remove this all this is the simple sir concentrate fermentation broth is the xylitol and then add the some dilution and then calcium carbonate add and calcium hydroxide addition and carbon dioxide gas sparging in this carbonation process and remove all the impurities and successfully approximately 70% हमें xylitol जो मिला इस process के through impurities मिली. Now is the second is the this is the summary of the कि initial जो हमारा था करीब जो loss था xylitol का इसमें very less लॉस था और जो अदर इम्प्योरिटीज थी जो जैसे अनवांटेड जो आपकी शुगर्स थी जो यूटिलाइज नहीं हुई फर्मेंटेशन में नॉट यूटिलाइज और जो अदर पेप्टाइड्स जो एमिनोगेम्स थे वो इजिली रिमूव हुए बाय यूजिंग द कार्बोनेशन और आयन एक्सचेंज प्रोसेस में. But 70% of xylitol is how to convert into 90% of the xylitol. This is a very difficult. We are using the activated charcoal. Activated charcoal is also further the remove of the phenolic content and successfully produce the xylitol syrup. And also they successfully we can xylitol syrup approximately 96% of the pure xylitol is produced. Now this is the our final process कि spent biomass reduce किया नाइट्रिक एसिड से किया लिक्विड सॉरी पार्ट्स को हमने कंटिन्यूअस सेंट्रीफ्यूज किया अल्ट्रा फिल्ट्रेशन फॉर द रिमूवल ऑफ द लार्ज मॉलिक्यूलर वेट एंड अदर्स जो प्रोड्यूस एसिड हाइड्रोलिसिस में हुए उसको हमने रिमूव किया सक्सेसफुली शुगर को हमने जाइलोस शुगर को हमने नैनो फिल्ट्रेशन से कंसंट्रेट किया बिकॉज जाइलोस शुगर का मालिकुलर वेट इज 150 मालिकुलर वेट एंड नैनो फिल्ट्रेशन कट ऑफ वैल्यू हमने 100 डाल्टन की रखी जिससे आपका ना जाइलोस इज कंसंट्रेटेड बाय नैनो फिल्ट्रेशन देन removal of the salt by using the ion exchange process and this fermentable sugar is converted into the successfully the xylitol and continuous nanofiltration then carbonation and exchange से हमने xylitol process किया सर and thank you sir any question please sir.
Thank you, sir, for delivering such wonderful talk on xylitol production and optimization. Sir, just I want to know one question from your side. You have mentioned initially. You have mentioned that lignocellulosic waste from aromatic plant is quite different from normal lignocellulosic.
Yes, sir.
So why you have selected the waste generated from aromatic plant for production of xylitol?
तो सर एक्चुअली जो लिग्नोसेल्यूलोसिक बायोमास है जैसे राइस स्ट्रॉ है कॉर्नकॉब है या अदर्स जो होते हैं तो सर हमने फोकस किया था हेमिसोलूस क्योंकि जो लिग्नोसेल्यूलोसिक बायोमास है जैसे राइस स्ट्रॉ है या कॉर्नकॉब्स या अदर्स जो बायोमास है वी आर वर्किंग तो उनमें हमको जो हेमिसिलोस पार्ट था उसमें एसिड हर्बिसिस करनी पड़ रही, मतलब हमने एसिड इसमें भी किया लेकिन उतना कंसंट्रेशन में हमने इसमें नहीं किया, मतलब जो लिग्नोसेल्यूलोजिक और जो एरोमेटिक स्पेंट बायोमास में क्या कि ऑलरेडी वाटर से डिस्टिलेशन हुआ, जो हाइड्रोथर्मल रिएक्शन हुआ, जो हाइड्रोथर्मल उसमें जो डिस्टिलेशन हुआ वो ऑलरेडी एक माइल्ड आपका हाइड्रोसिस हो गई। तो उससे क्या हुआ कि सर कि जो इनहिबिटर्स थे वो मैक्सिमम तो आपके पहले हाइड्रोथर्मल में ही निकल गए और वो इजिली जो बाउंड था, जो लिग्निन कंटेंट से जो टाइटली बाउंड थे वो लूज हो गए ड्यूरिंग द फर्स्ट डिस्टिलेशन प्रोसेस में। तो और दूसरा थिंग थी कि एरोमेटिक वेस्ट में जो इसमें सेलुलोज हेमिसेलुलोज कंटेंट था वो हमें अच्छी तरह से मतलब 25 to 30% हमें मिला।
Okay, audience, if you have any question, you can ask your question or queries to Dr. Vinod. Okay, no more question. Thank you, sir. Thank you so much. On the behalf of this conference, I am thankful to you for joining
Thank
this.
you, sir. Thank you, sir.
With this, we conclude our this session. Please put up your hands for the wonderful talks in this session. We sincerely extend our deepest thanks to all our speakers. And let's do the felicitation now. So may I request our session chair, Dr. Santosh, to please come forward. And our speakers, since two of them joined online and were already there, so we request Dr. Nidhi ma'am to please come forward and receive your award. May I also request Nutan Kaushik ma'am to please come, please, sir, and present her the memento. Also requesting Bandyopadhyay sir.
Please put your hands together.
We would also like to thank our gifting partners, Unibit and Ayuth.
Now we will also request Sir.
We request Bandyopadhyay sir to be there only because we would now like to felicitate.
We will kindly honor our chair and co-chairperson as a token of our gratitude and appreciation for guiding us through the whole session. We request Dr. Bandyopadhyay sir and Nidhi Chaudhary ma'am and Nutan Kaushik ma'am. Thank you. Thanks a lot, sir. Please be seated. We will not have a tea break. We will start with our next session. It is my privilege to welcome all to the session on plant and agricultural biotechnology of the prestigious Conference on Sustainable Biotechnology and Bio-innovation 26. Plant biotechnology has emerged as a transformative force in addressing global challenges such as food security, climate change, sustainable agriculture, and nutritional enhancement. From the genetic improvement to precision breeding and climate resilience crops, this field continues to redefine the future of agriculture. Today's session will explore innovation research, sustainable strategies, and technological advancements that are shaping modern agricultural systems. It is our honor to introduce our first session chair, Dr. D.K. Bandyopadhyay. Dr. D.K. Bandyopadhyay is a scholar, academician, and administrator, also a visionary leader in India's education community in modern times. He is a gold medalist in his master's degree and has 2 PhDs, one in computer application and another in management with inputs from law. He was the Dean and Acting Director of IIM Lucknow, Director of Indian Institute of Forest Management, Bhopal, Vice Chancellor of Guru Gobind Singh Indraprastha University, and was also the Vice Chancellor of IMS University, Dehradun. Presently, he is the Chief Advisor to our Founder President and Chairman ALS. He has been in different committees of Government of India, such as Chairman, Consultancy Development Center, CDC, Chairman, Technical Executive Committee for Access to Knowledge, DSIR, Government of India for Technology Development, Member General Council and Executive Committee for National Accreditation Board, New Delhi, Chairman, Finalization of Indian Standard on Educational Services Bureau of Indian Standard, Member Executive Committee of AICTE, Member Curriculum Review Committee, NIT Patna and NIT Durgapur, Member Secretary for Evaluation of National Mission on Education through ICT Program, MHRD, Government of India. He is a member, Board of Governor, NIT Hamirpur, Council Member, All India Management Association, Chairman, NAC Committee for Accreditation of Different Academic Institutions. He is also a recipient of various awards such as Kiva Noira Award for Academic Leadership in Management Education by AIMA 2010, Faculty Gold Medal for Excellence in Academic Achievements, Slovak University Nitra 2010, Guru Vaishta Award by Scholar Society 2009, Delhi Ratan Award 2009, Best Dedicated Professor IIM Lucknow Award 1996, Indira Gandhi Priyadarshini Award 2010, Dr. Radhakrishnan Memorial National Award 2011, Mary Dilli Award 2012, Top Rankers Excellence Academy Leadership Award 2013. So these are few I think we have mentioned, and there are so many awards, recipient of so many awards. It's our privilege, sir, to have you on this session, and I humbly request our distinguished chairperson, Dr. D.K. Bandyopadhyay, to please take the seat on the dais and preside over the session.
Our co-chair for the session is Dr. Kishore Kumar Krishnani. Before I invite him on the dais, may I just run through his brief bio sketch? Dr. Krishnani is a Joint Director of Academics at ICR, Indian Institute of Agricultural Biotechnology. He possesses 32 years of experience with 6 ICAR and 6 overseas universities in the field of environmental biotechnology. His research focus has been on the development and application of bioanalytical and molecular techniques, including metagenomics, bioremediation, nanobiotechnology, hydrogel-based microencapsulation for monitoring and alleviation of climate change-induced abiotic and biotic stresses, input use efficiency, and examination of uncultured bacterial diversity. He has over 200 publications, including 115 journals with 6,520 citations, a 39H index, is released 350 gene sequences and 12 patents and technologies. He has imparted research-driven teachings and as a major advisor, guided 8 PG and 3 PhD students. He has received several awards, including ICR's Rafi Ahmed Kidwai, NAS Biennium Recognition, Fellow of NAS, Fellow and Member Board of Governors BRSI, BHU Centennial, Member NASI, Australian Government's Endeavour Fellowship, a trusted mentor, ICR NAIP Overseas Fellowship, and DBT's Biotechnology Overseas Associateship at USA, IFF Young Scientist, GCA's Fellowship at Philippines, Best Presentation Awards in recognition of his outstanding contribution in environmental biotechnology. With his glorified biosketch, may I request Dr. Krishnani to please chair the session alongside Dr. Bandyopadhyay. Please put your hands together and welcome him. So we're truly honored to have them guiding this session. We request all the speakers to kindly adhere to the timelines. Thank you.
I must congratulate MIT Institute of Biotechnology for organizing such an eventful conference with an eventful, well-recognized topics. Now, today's topic, if I just look at it, land and agriculture biotechnology. And within that, there are 4 subdivisions, as already mentioned. I don't want to repeat them, but certainly the first one, bio-innovations for sustainable agriculture and food system. Who other than Dr. Nutan Kaushik can give the justification for that topic? Now, those who do not know Dr. Kaushik, those students who do not know Dr. Kaushik, I shall give just brief outline about her achievements. She is PhD in agriculture, agricultural chemicals from Indian Agriculture Research Institute, and extensive experiences of over 35 years. And now I am just shortcutting it. You see the well-traveled persons and also achieved so many accolades. Whenever we visit to any other country who are associated with agriculture or agricultural system and bio-innovative sustainable agriculture, you name Dr. Nutan Kaushik, but yes, we know her. And then I personally get recognition through her recognition. And that is the type of person she is. And her mainly, the contribution is that standardized half seed method for estimation of fatty acid for brassica seeds under chemotyping activity. Her efforts has led to the identification of several chemotypes of neem, barberries, oseam, asparagus, chlorophyllum, Withania, Tylophora, Karanja, and so on and so forth, which are high oil content and isolation of new bioactive compounds. She has worked as a visiting scientist in several international universities in USA, Germany, France, Australia, Spain, Norway, as I was mentioning, Tunisia, Netherlands, as visiting scientist. She was USDA Fellow at Michigan State University, USA, Nuffic Fellow at Wageningen University, Netherlands, DBT Crest Fellow in University of California, DAD Research Ambassador. She has handled more than 50 projects as PI or co-PI, including 18 international projects of EU, USAID, and UNEP. Indo-German, DAD, Indo-French, Indo-Australia, Indo-US, Indo-Spanish, Indo-Norway, and Indo-Tunisia, and she has traveled widely with 35 countries. She has published more than 90 papers in reputed journals with combined impact factor of more than 500 and filed 27 patents. developed several technologies, and she has commercialized 3 Indian out of that and 1 US company. The Bio-innovations commercialized to Spanish company, developed several variety of Branchia, Junecia, and B. nepos. I'm sorry that this terminology is something Anyway, microbial antifungal formulation, high processing waste valorization. This is most importantly, high processing waste valorization into protein, hydrostate, and gelatin, functional food, protein enriched soup, metabol technology for introduction of agri-food production by artificial infection, IPM packages, climate smart agriculture, and so on and so forth. And she has been awarded Best BIRAC project by BIRAC, DAD Research Ambassador 2010, Bharat Jyoti Award, and more recently, Bio Agriculture Asia Award 2022, Special Achievement Award. She has been recognized as she is top 75 chemists in India by Royal Society of Chemistry, UK. She has also been awarded various prestigious international fellowships like USDA, then UNESCO Fellowship, DBT Crest Fellowship, and she has served as a member of the Expert Review Committee and member of Task Force of BIRAC and DBT. That's just a few introduction of hers, but her major introduction is that we get recognized by her name. That's the most important thing. Now, may I request Dr. Nathan Doshi just to talk about bio-innovation for sustainable agriculture and food system.
Thank you, sir. I think would you mind if I talk by hand mic? Okay, thank you. Very good evening to all of you, and thank you so much, Dr. Bhadrapadhyay, sir, for metaphorically introducing me. Thank you, sir. Hello, students. Can I have your attention, please? Okay, please. आप आगे आ जाइए, please. सब लोग आगे आ जाइए. इधर आ जाइए. ये सारी कॉन्फ्रेंस आप ही के लिए हो रही है ना? Please come forward. Please come forward and sit here. I'll come down. कुछ है इसको पॉइंटर जिसमें स्लाइड्स में मूव कर सकूं? It's okay. It's okay. It's okay. It's okay. It's okay. Please. Quickly, come here. This table is free. टेबल पे आइए आप प्लीज. आगे टेबल पर आइए. सब लोग बच्चे आ जाइए. All good. Okay. So I am talking about the bio-innovations for sustainable agriculture food systems. You see this slide? How many of you know about the Green Revolution? Raise your hand. Green Revolution. Green Revolution. How many of you know? Green Revolution. You students, please see this slide. How many of you know? You know Green Revolution? Okay. Who raised the hand? Okay. What is Green Revolution? Okay. Do you really know how much we were producing at the time of independence? No. Do you know how much we are currently producing? Any idea? Any guesses? When we talk about the agriculture production, we only talk about the cereals. We don't talk about the pulses, oil seeds, because when we talk about the food security, we talk about only 2 crops. We talk about only the cereals. Do you know what is the difference between the cereals, pulses, and oil seeds? You know? Okay. So when we got independence, at that time our food production was only 50 million tons. How much? 50 billion tons. and we were importing food from USA. वो PL80 नाम सुना है कभी आपने? PL80. We used to import PL80 from USA, and that was such a bad wheat, that bad quality wheat, very bad quality. आपने तो सर देखा ही होगा. And people used to have that too was available in ration. You're very fortunate that there are no more ration lines now. It all happened because of green revolution. So from 50 million tons, today we are producing more than 350 million tons. How much? 350 million tons. Can you tell how many times we have increased this production? 7 times. Good, very good. So almost 7 times. We are talking only about the food cereals. We are not talking about the milk or fruits and vegetables and or many other things in which we have gained the surplus. Now we have become the surplus producing. It all happened because of the green revolution. And what was the green revolution all about? It was all about introducing the technologies to the farmer. What was that? Technologies to the farmers. What were those technologies? Do you know? Have any idea? Any idea what technology must have contributed to the significant increase in this production? We are talking about the bio-innovation, no? What are bio-innovation? They are all technologies. Okay? So, GMO नहीं था बेटा उस समय. GMO अभी आया है. GMO तो अभी आया है. Yes, seed is one. Hybrid seeds. Okay? Hybrid से हमने productivity बढ़ाई. First thing. Second, what is the second technology which contributed significantly? Irrigation. Okay? You know, irrigation alone can increase the production 3 times. 3 times. Only irrigation. Rain-fed versus irrigated. There's a tremendous difference in this thing. Secondly, the short duration varieties of the wheat. The biomass production was less. The yield was higher yielding varieties, and that too hybrid varieties. Second thing, third thing, pesticides. We introduced pesticides to control the pest and diseases, and the fertilizer to main pest the fertility. That was very good. So that was very good until unless farmers started misusing these inputs. What are these called? These are called the inputs. Inputs for the farming inputs. Seed, fertilizer, pesticide, water, all these are called the inputs. So when farmers thought, okay, by applying 1 kg of urea, I can get the double of the yield. Why I should not apply the 10 kg of the urea? But that was the injudicious use. What was that? Injudicious use. आप नमक थोड़ा खाते हैं ना खाने में. अगर पूरा कूप डाल 1 कप नमक डाल दोगे अगर अपने खाने में, can you eat it? Can you? It will become poisonous. Huh? The same salt which you have in a small quantity will become poisonous if you add more. Same thing happened with these inputs also. So when they started judiciously using these inputs, they started having the bad effect on the soil health, on the air, and the water quality, and the food quality. Everything started deteriorating. So that's why we are not talking about the evergreen revolution. So the green revolution, father of green revolution, is written here. Who is the father of green revolution? M.S. Swamy. So that's where in 1968, after the independence till 1968, we had the green revolution. Then came the white revolution. Do you know what is white revolution? Milk, yes, definitely. Milk production. So in till 1970, we came surplus production in milk production. Then came the information technology in 1984. Then came the blue revolution. How many of you know blue revolution? Blue revolution. Blue revolution. Huh? Fisheries, yes. The blue revolution is all about marine and fishes. Then came the biotechnology. Now, you are all the students of biotechnology. This came somewhere in 80s, late 80s, 1980s. So we have the biotechnological revolution. And then yellow revolution. Now we are talking about the yellow revolution. What is yellow revolution? Oil seeds. Very good. Excellent. So all about the oil seeds. And then now we are talking about the evergreen revolution. What does the evergreen revolution mean? The revolution which does not have deteriorate the environment, increases the yield, maintains the yield, increases the yield, but does not deteriorate the environment, soil, does not pollute our natural resources, and does not pollute in our air, water, anything, or does not it does not produce a poisonous food. So that is where we are now talking about. And that so whatever the bio innovations we talking about should cater to the evergreen revolution. So now, is this 350 million enough for us? Should we stop? Should we stagnate the yield? No. Why? Why? You see here, the 3 billion, we have to cater the growing population. We have to cater to the growing population. Population is growing, demand is growing. So we have to produce more from less land. Land is decreasing. Per capita land is decreasing per capita land. I'm not talking about the land mass. I'm talking about the per capita land. Per capita land is decreasing, and so we have to produce more from the same piece of land. Okay? That is one thing. Another interesting figure is this one. Do you see this one? What does it depicts, this particular figure? So it was that it is one farmer catering to how many people over the period of time. Okay? Starting from 50s. So in 1950, one farmer was catering to 15.5 people. But by 2020, how many? 200. Same farmer. So one farmer is catering to 200 people. So the yield, definitely the yield has to go up. And on the top of that, what does these figures indicate? CO2, methane, N2O, climate change. One word is climate change. So these are all about climate change. You see the pre-independence era values versus the values in 2019. There's a significant increase in the emissions. We don't say production. What the word we use is the emission. There's an increase in the emission of the CO2, methane, and N2O. Okay. So this is how the climate is changing. The gases, as the gas emission is increasing, the temperature is increasing because of these emissions. So what is the impact? So this is the impact of the climate change on agriculture. Very obvious. This changes in water supply and demand. How the water supply and demand is changing because of the climate change? Because erratic rainfall, erratic precipitation. Okay. And so the water supply is changing. In some way there's a flooding, in some way there's a drought. So these are the changing, the scenarios are changing. Secondly, the significant impact on crop yield. High temperature. अभी देखिए इस महीने में आपको टेम्परेचर कहाँ पहुंच गया? How much? You're feeling the climate change, isn't it? So there's a sudden increase in the temperature will yield to the crop loss, because the crop will start flowering immediately. In the distress condition, what the plant does? You are a plant biotechnology students. The plant in the distress condition, the plant started bolting, the flowering, everything happens. What it has to survive? It has to give the offspring. So in whatever the photosynthetic savings are there, all that saving goes into the flowering and then seed production. So seed production is hampered because of that. Then changes in geographical distribution vigor of insects. What does it mean? It means the insects who were never visited now, you see the mosquitoes in Himalayas also. Why? Because the temperature is increasing. So if there's an increase in temperature, the insects will also change their territory. They will move upwards. They will move upward and they will start impacting your agriculture. Then environmental instability may reduce the effectiveness of the pesticides. High temperature, the effectiveness may go down. That growing threat of food insecurity and malnutrition, of course, the food quality and the food quantity, both are impacted by increase in temperature, and that's so both are impacted. Okay. So what are the other global challenges? The other global challenges the food system contribute to. Food system also contribute to the greenhouse gas emissions, nearly 31% are. Then 33% of the soils are degraded. I told you about the redevolution. And because of the overuse of the water, overuse of the pesticide, overuse of the fertilizer, the soil degradation has happened. So now what we have 33% of the soil degraded. Okay. And then 828 billion people face hunger. They go hungry. Climate variability threatens the crop variability. So when we are talking about the bio-innovations, isn't it? This session is all about bio-innovation. So what innovation you should do? Looking into these problems, what are innovations? Why we do innovate? Why we innovate? First of all, why do we innovate? We do innovate to solve a problem, isn't it? Why the innovation is done? It's just a kind of play toys or something? No. You do innovate to solve some. Yeah, that would be fine. Okay, sir. Thank you. Okay. So this is how you do. I will just skip this slide because I already introduced this. Soil health, I already introduced. Rising. So innovations we are talking about. So bio-innovations, application of biology. As the word says bio, you have to use the biology, isn't it? So anything dealing with biological system and innovating a new product is a bio-innovation. So why do you innovate? You innovate to reduce the environmental footprint. Any innovation which can reduce the environmental footprint and enhance the productivity resilience. So these are the two major objectives when you do the bio-innovation. So what are the technologies which you can bio-innovate? How many of you know microbiology? I think most of all of you must. Okay. So what are the microbiological solutions for the sustainable agriculture? PGPR, what is the full form of PGPR? PGPR. Very good, excellent. So PGPR are the microbes which can enhance the plant growth. Then rhizospheric engineering. You can do the engineering of the rhizosphere by having the microbial system. And then mycorrhiza for phosphorus globulation, then nitrogen fixing, biofertilizer, and the biocontrol and microbes. So these are some of the solutions which you can give as alternative to fertilizer. These are the biofertilizer or plant growth promoters which can help the growth of the plants, which you can develop. Then we have biopesticides. So have you heard the word biopesticides? How many of you? Biopesticides? Okay. So these are the biological way of controlling the insects. So it could be a plant, it could be fungi, it could be a virus, it could be bacteria, it could be a mycoplasma also. So anything which controls the insect can be converted into a product, and that's a biopesticide. They are best. So most popular ones are the Bacillus, Trichoderma, and Pseudomonas. These are the three most popular commercialized biopesticide which are available. But do you think these three are sufficient to control all the insects, all the problems? No. So you need to innovate more. to control more diseases, to control more insects. Okay. So there's an arrow. How many insects are there? 1,000, more than 10,000 insects. But these three cannot control all the 10,000 insects. Similarly, million of microbes which are pathogenic in nature. To control that, you need a specific monster. So there's a plenty of scope to develop the biopesticide for controlling different diseases. Then biostimulants. Have you heard the word biostimulants? Biostimulants? No. Biostimulants are the one which stimulates the plant growth by signaling, producing the molecules which triggers the genes inside the plants. So just like the immune system, no, they enhances the plant immune system. So they give the signals by which the plant immune system is enhanced. So now a lot of discussion is taking place about the biostimulants, and biostimulants can come from seaweed extract. You know, seaweed extract is a biostimulant. Humic acid is another biostimulant. Amino acid formulations are another classes of biostimulants. Then stress tolerance enhance. So they enhance the tolerance within the plant system, just like our churnprash. Okay. Then precision bio inputs. So you can have the drone-based delivery system, the AI advisory system, variable rate application, and reduced wastage. Then genome-enabled breeding. I think in the previous session, somebody mentioned about it, the genome editing and everything. So we can have you can breed the varieties which are tolerant to insect, which are tolerant to diseases, or which are higher yielding, or which are higher in quality. So these are the 4 targets which you can have for the breeding and then for biofortification. So what are the ways by which you can do? Markers-assisted selection, genome selection, CRISPR-Cas editing, or climate editing cultivars, or just tapping the biodiversity and developing the cultivars. So biofortification, just to enhance the quality of the produce, you can do the biofortification. Zinc and iron are the most prevalent. If you are deficient in zinc, you will have a lot of health problems. Our soil is deficient in zinc. If there's nothing in the soil, you cannot have in your food. If it's not in your food, you will not get it. So there's a concept called One Health. One Health means if your soil is healthy, you are healthy. So you have to make the soil healthy. And if it is not, you have to make the food healthy. Okay. Then circular bioeconomy. What does it mean? It means if you're in a production system, the byproduct of a production system is the raw material for another product. Okay. So you are using, we call it rest raw material. We don't call it byproduct. We don't call it waste. We just call it rest raw material. Means there's a raw material. You're taking one portion of the raw material for producing certain items, certain goods, certain food products, or certain products. But rest of the raw material is still available for developing some other kind of product. So that's how the circular economy works on. That so we have to, for the valorization of the agri residue, food processing waste, bioenergy, and composting and biochar. These are all applications of the circular bioeconomy. Then building climate resilience in crop. I think there are various stages. I will not discuss in length because the time is short, but you can go through my PPT, and these are the different ways for different hotspots. Hotspots are what? Like there are certain areas which are drought tolerant, drought prone. There are certain areas which are flood prone. There are other areas which has high temperature. Based upon the area, you have to have the strategy to tackle the climate change. You can see this in my paper, one of my paper. Then for carbon sequestration, biochar is a very good candidate, which can, and then reduce emission by having this, by manipulating the practices by cultivating the rice, and also by manipulating the food of the cattle. And then, so that goes to the climate smart agriculture. Then post-harvest bio-innovations, we can work on the biopreservations. You can have the biological preservation rather than the chemical preservatives. Then edible coating by enhancing the shelf life. You know the food loss contributes to a lot of greenhouse emissions. And food loss also contributes to loss of our natural resources. So that way, if you can minimize the food loss, you can do a great deal of contribution. Then biodegradable packaging is another way by which you can do the post-harvest innovations. Then packaging, we have waste to board. Then integrated food system approach is the one where you have the soil health management, digital plus biological integration, the biological innovations coupled with the digital application can do wonders in the field. And these things are happening in the field. Now sustainable intensification will be the key for this one. So you have to align the policy. All these things, bio-innovations, which you will dealing with this, all this for the environment, tech are these directly with SDG 2, which is for the zero hunger, SDG 12, that is sustainable development. SDG is sustainable development goal. How many goals are been defined by the UN SDG? How many SDGs we have? How many SDGs? 17. Very good. Excellent. Good. So out of 17, 3 are directly dealt by the agriculture. Then what are the future trends? Synthetic biology is one of the answers where the bio-innovation can happen. Microbiome engineering is another one where you people can innovate. This is basically for you this slide. Okay, where to do the NTCC? Huh? This is an NTCC slide. Okay. Carbon credit agriculture and then AI integrated farming. How to integrate the AI and biotechnology together to do the farming. Okay. So what are the key takeaways? Bio-innovation, enhance sustainability. Nobody can doubt about it. Improve resilience, reduce chemical dependency. That's the objective. And then enable circular economy. If we meet all these objectives, I think your bio-innovation will be a sustainable bio-innovation. Thank you. Thank you so much. I've not taken much time. Any questions? I think they have any burning question. No. Thank you.
So after this wonderful talk by Dr. Nutan Kaushik, now we have invited talk to be delivered by Dr. Sunil Kaul, National Institute of Advanced Industrial Science and Technology, Japan. We welcome Dr. Sunil Kaul. Dr. Sunil Kaul is an invited senior research scientist at National Institute of Advanced Industrial Science and Technology in Japan. His primary research focuses on uncovering the molecular mechanism behind stress, aging, and cancer. He has 300 publications in a high visibility journal, and he's the editorial board member of multiple scientific journals. He has received several honors and awards, including Fellow of FGSI, Overseas Fellow of BNSI, Fellow of Indian Academy of Neuroscience, Foreign Fellow of National Academy of Sciences, that is NACI. And additionally, he's a founder and president of Cold Tech Company Limited in Japan. So he is going to deliver a talk on novel ashwagandha extract, medicinal aromatic plant. I hope it's going to be very interesting, right? Medicinal aromatic plant for sustaining quality of life in an era of environmental stress. So online presentation. So let us welcome Dr. Sunil Kaul. Dr. Sunil Kaul, please.
Thank you so much. Am I audible?
Big applause, please. Yeah.
Am I audible?
Yeah, you are audible. Yeah. Please
yes.
go ahead.
Okay. Yes. So first of all, I would like to thank the organizer for giving me this opportunity to share our work with you all today. So as the title goes, I will be taking through our work on ashwagandha and try to convince how different extracts help in sustaining quality of life in an era of environmental stress. So, yes, as we all know that stress, aging and cancer are closely connected. As we age, the number of incidents of cancer increases, so it is very apparent at the age of 50 or 55. Likewise, in culture, cells grow for certain population doubling, after which they age and die. But if something goes wrong with the one cell, it keeps dividing and become immortal or cancerous. So in our lab, we want to understand why normal cells age and why cancer cells keep dividing. See, in other words, we were looking for some factors, molecules that could enhance the lifespan of normal cells or inhibit the growth of the cancer cells, that is cancer therapy. So we tried doing screening of several phytochemicals and then fixed on ashwagandha, that is Vidania somnifera, for the reasons that ashwagandha has been used in Ayurveda for hundreds of years for increasing memory, treating insomnia, stress, hypertension, diabetes, arthritis, and so on. But the mechanism was not known. So we wanted to see if ashwagandha actives be used for the intervention of cancer and stress, because our background is on molecular mechanism of stress, cancer, and aging. So what we did, we made a ashwagandha leaf extract for the reason that we can have ample supply without sacrificing the plant. And then we made an ethanol alcoholic extract, characterize all the components present in the extract, and put them in our in vitro culture system, where we always compare normal and cancer cells in parallel. We want something which can kill cancer cells, but it is non-effective to normal cells. Once we find the candidate, we move on to in vivo system, where we use rodents. So what we found, that our initial observation was that ashwagandha leaf extract, when we treat the cancer, it kills, but normal cells were spared. So in this cartoon, you can see that here are normal cells, cancer cells. We are treating with ashwagandha leaf extract to cancer. Only cancer cells are dying, but not normal cells. And another encouraging observation was when we inject the nude mouse with the fibrosarcoma, very active cancer cells, they make a very solid tumor by day 12. But in parallel, if we inject the alcoholic extract, then there is a strong inhibition of tumor. That means ashwagandha leaf extract, eye extract, is strongly inhibiting the tumor growth. So we went on to find the bioactive compound in this extract, and then by HPLC, we found that this is one of the compound is vidanone, the others are vidafrin A and other vidanolites. So by NMR, we confirmed that this is vidanone. So after having all these vidanolites in hand, we went on to check which ones are very inhibiting the strong tumors and others are not. So we find that ashwagandha leaf extract is strongly inhibiting the tumor, but other vidanolites like vidanolites A, deoxyvidanolites are non-active, whereas vidafrin A is strongly inhibiting the tumors. So now we moved again back to in vitro system and then compared normal cells and cancer cells in parallel. Here we are taking bone cancer and breast cancer cells, and then to compare, we are taking normal fibroblast. What we see that when we treat the cancer cells, there is a strong killing in cancer cells, both the cancer cells, but nothing happens to normal cells with eye extract. But when we treat with vidafrin A kills cancer cells, but it also kills normal cells at that concentration. There are several vidanolites which is non-active, but interestingly, vidafrin A that kills normal cells, if we treat them with vidanone, there is a killing is reduced. So what I have told you so far, that ashwagandha leaf extract exhibits cancer inhibitory activity with vidanone as its primary active component. Now the mechanism, the actual next step is how to understand the mechanism, how ashwagandha extract is killing cancer cells. So we did 3 types of approach. One is a loss of function screening to analyze the selective killing of cancer cells. Here we played the cells in 96 well plates and transfect with ssRNA library in the presence of extract. So the extract should kill the cells, but because there is a ssRNA, there are some cells are viable. So we find the targets. Similarly, the second approach is to use randomized ribozyme library screening method, where we are first infecting the cells with the vector of ribozymes in the presence of extract. Extract kills cancer cells. But when we are using the library in the presence of extract, some cells are viable. We extract that and then identify the components. So whatever the compound we found from the first approach and second approach, we put them in the third approach, which is a bioinformatics approach. By this, we came to know that ashwagandha leaf extract kills cancer cells by two major pathways. One is p53, p21 pathway, the other is oxidative stress. Now the question is, we have to validate whether p53, because p53 is an important protein, whether it is really involved. So to do the validation, before that, I would like to introduce p53. p53 is a tumor suppressor protein which controls the cell division. In cancers, more than 70% cancers, it is inactive by either mutation or just inactivation. So we wanted to know how p53 is involved in this killing of cancer cells. So we did the biochemical analysis. Here again, we are comparing cancer cells, osteosarcoma, with normal fibroblast, treated them with Vidanone or I extract, ashwagandha extract, and then see the expression of p53. As you see in these cancer cells, there is a very strong activation of p53. Also, the downstream p21 is activated. But in normal cells, there is no change in p53, which complements our earlier observation that ashwagandha leaf extract kills cancer cells by activating p53, p21 pathway, but it is harmless to normal cells. Now, to further complement this our observation, we'll try to look at the p21 downstream protein. We again treated the cancer cells, breast cancer cells, MCF7, and compared normal fibroblast, treated them with Vidanone and I extract, and look at the p21 expression. We see here only in cancer cells, p21 is very well expressed. By immunostaining, again we see that p21, which is colored red here, fluorescence red, we see that in response to I extract, ashwagandha extract, there are a lot of R or red color, which is p21 expression in the nucleus only in the cancer cells, very few in normal cells. Similarly, in response to Vidanone, we find a lot of red color or p21 expression in cancer cells, but less in normal cells. So the other pathway, as I said, that it is by oxidative stress. To further analyze how ashwagandha leaf extract is killing by oxidative stress, we did ultrastructure study here. In control, we find that the mitochondria is very well defined, cristae are well placed, but after the treatment with Vidanone, mitochondria are disorganized and cristae are not in proper place. So to further complement, we also look at the ROS or reactive oxygen species in normal and cancer cells. So we took normal fibroblast TIG3 and compared with the breast cancer cells MCF7 and treated them with ashwagandha leaf extract. We see there's a lot of green color or the production of ROS in cancer cells, but not in normal cells. Similar results were seen in response to Vidanone in cancer cells, but not in normal cells. So what that means is ashwagandha leaf extract kills cancer cells by the mechanism involves activation of p53 or oxidative stress. A predominant active components are Vidanone and Vidaferin A. Now, ashwagandha kills cancer cells in general, but to be more particular, we made a nanocapsules, folic acid ashwagandha extract nanocapsules. The idea is these nanocapsules are loaded with ashwagandha extract. As it comes to the membrane of the folic acid receptors positive cells, it goes inside and then the extract is released and it kills cancer cells. So to find the cells with folic acid receptor positive cells, we did the biochemical analysis, and as we see here, the HeLa cervical cancer has a very strong folic acid receptor. Bone cancer also has a folic acid receptor, but breast cancer or other cancers or normal cells don't have these folic acid receptors. So that means this ashwagandha nanocapsule that we made will specifically go inside HeLa cells or U2S, but not in normal or other cancer cells. So to prove that, we again did a nude mice assay. Here we have injected cells with folic acid receptor minus cells or positive cells. So as we see here, folic acid receptor cells, when we treat with eye extract, it slowly induces or inhibits the tumor in a very systematic way. But when we are using nanocapsules with ashwagandha in folic acid receptor positive cells, there is a very drastic reduction of tumors. So that means folic acid receptor ashwagandha nanocapsules specifically target folic acid receptor cancer cells. So we are here very precise. Now this is a story when we are using the alcoholic extract. So you want to ask me what happens if we are using water extract. For example, you want to have ashwagandha in water. So we went on to see by doing battery of experiment that we did with the alcoholic extract, so in water extract, and then again the analysis, and this is one oh okay, so this video is not working anyway. So here in this guy rats, we have stereotaxically implanted C6 cells into the rat brain, and this rat has a big tumor on the right hand side and it moves on the right. But if parallelly, if we feed this rat with the water extract of ashwagandha, there is a much normalcy. So when we sacrifice these guys and then look at it histologically, we find a big tumor in this C6 cells implanted brains of these rats. But after the feeding with ashwagandha extract, there's a very reduction in the tumor, which can be seen by fluorescence cells, marked for fluorescence cells. And then we wanted to see whether this water extract also had vidanone or vidafurin A. But to our surprise, we found that in this water extract, the active component is triethylene glycol, small molecule. And the pathways by which water extract kills cancer cell is not by P53 or P21 pathway, but it takes a PRB pathway, retinoblastoma pathway, which is an alternative pathway in cancer treatment. So that means water extract also selectively kills cancer cells. A main active ingredient is triethylene glycol, and mechanism involves activation of retinoblastoma protein. So what that means is ashwagandha leaves we have made alcoholic extract, water extract. Alcoholic extract has two main components, vidanone and vidafurin A. Water extract has triethylene glycol. Alcoholic extract kills cancer cells by activation of P53 or by oxidative stress, but water extract kills by activation of retinoblastoma protein. So if we take ashwagandha as such, then both these pathways are activated and we can do nice tumor management. This is the reason why in Ayurveda always said that to take herbs as such and not to disintegrate or separate them in any compound or any form of extracts. Now, after knowing that vidanone and vidafurin rich is important to make ashwagandha active for killing cancer cells, we went on to make different innovation. The first one is to see if we can make extraction rich in this vidafurin and vidanone. So to do that, we took the help of cyclodextrin in collaboration with the cyclochem company. The cyclodextrins are naturally derived from starch or glucose polymers. They have a circular structure. The inner wall contains a dense arrangement of hydroxyl groups and is hydrophobic, while the outer wall is hydrophilic. So we used all the three alpha, beta, gamma type of cyclodextrin and then see if we can enhance the yield of vidanone and vidafurin A in the extract. And indeed, we found that some of the extract used in some of the cyclodextrin indeed has more vidanone and vidafurin A. And these extract are more active, and less amount of extract could be used to kill more cancer cells. Next, we wanted to make ashwagandha that has more of these vidanolites or vidanone and vidafurin active compounds. So in Japan, we are making organically grown ashwagandha. We are also making ashwagandha growing in greenhouse or using vermicompost. and we have been successful in uh, cultivating ashwagandha in hydroponically in water, and
May I request to just wrap it up?
yes, please. Yes, so this is the last slide. So we've been successful in making hydroponic for the reason that we can grow ashwagandha without any external contamination and in a very similar pattern. So these are the books we have produced: Science of Ashwagandha Preventing Therapeutic Potential and Ashwagandha for Quality of Life. With this, I would like to stop and acknowledge all my friends, students, researchers who have contributed to this work, and collaborators: Dr. Sundar from IIT Delhi, Dr. Gurcharan and Dr. Pati from Guru Nanak, Professor Redl from CMRI, Dr. Chiok from Hanyang University, and Dr. Therao from Cyclochem for collaboration. Thank you for your attention.
Thank you, Dr. Sunil Kaul, for
Yes.
very informative talk. Your presentation on extract for this novel ashwagandha extract for sustaining quality of life in era of environmental stress was really very much useful for our student and the faculty. Thank you very much.
Thank you so much.
Since we are running short of time, so we can take one or maximum two questions. Yeah. I'm having them. Dr. Sunil, nice presentation.
Yes, thank you.
Mentioned about the MCF7 breast cancer cells that you have used. Have you
Yes.
looked for the TNBC also?
No, we haven't.
Another important aspect is that you have mentioned about the leaf extract.
Yes.
Majority of the alkaloids are there at the root of the
Yes.
ashwagandha.
Yes.
So have you looked into that also?
Yes, we try to compare root, stem, leaves to begin with, and we find that this vidanone and vidaferin, which is active and which are good for cancer, is mainly found in leaves. So we focused on leaves. So there are about 20 times more vidanolides in leaves than roots. That's why we focus on leaves.
Thank you.
Thank you.
Hello. Hello, Dr. Kaul. I'm Nutan Kaushik this side.
Sure.
I have one question that
Sure.
you said that ethanolic extract has different mechanism and water extract has different mechanism of
Yes.
controlling the cancer cell lines. Did you try the aqueous alcoholic extract also?
Alcoholic extract?
Aqueous alcoholic extract.
No, we didn't do it. We only do it.
No, aqueous alcoholic extract.
Aqueous alcohol. We didn't do it. We only tried.
We may try that also.
Okay, I will. Yes.
That way, maybe in single extract you may have both the activities together.
Yes.
Thank
you so much.
Dr. Kaul, I have got one question.
Sure.
You see, now ashwagandha is now being researched and also used for a long time in India. But it has been observed that naturally produced ashwagandha and then in a artificial way of producing ashwagandha, the properties get drastically changed.
Yes.
Have you taken note of it?
Yes. So we compared ashwagandha growing outside in organically and in greenhouse, hydroponically, aeroponically. We compared all, but for clinical trial, we have to have a very pure ashwagandha. So that's why we moved on to now hydroponically, and we have also gone through the clinical trial and completed a phase 1 clinical trial in USA recently. So moving now to phase 2 with our alcoholic extract.
Dr. Kaul, I have a small query. This
Sure.
ethanol, since it has asymmetric carbon atom, so
Yes.
it has that enantiomers, stereoisomers, and there is a
Yes.
possibility that one stereoisomer may be active and the other isomer may not.
It is possible.
It's possible,
Yes.
right?
Even the vidanone, which is found in ashwagandha in different from different sources, there's a conformational change. So there is a possibility we have to check every time whenever we use ashwagandha. So all ashwagandha are not same, all are different. So we have to have a very, very specific and cloned ashwagandha for our use in clinical studies. So this is the lacking information that all ashwagandha are not same.
I think Dr. Sunil, you can contact we have a medicinal aromatic plant institute in Anand, ICR Institute. So you
Yes.
can talk to some scientists in that institute. No, probably they may be having some information related to this. So thank you very much.
Thank you so much. Yes, thank you.
I think one more question.
Yes, sure.
As a small observation, Dr. Kaul,
Yes,
I have
please.
done the chemotyping of the 167 accessions of ashwagandha collected from India. And as you
Yes.
said, all are different. So you must define the chemotype which is used for your studies here.
Yes, we have now clone that is good for one particular type of cancer or different clone for different cancers. We already have clones now, already chemotyped, and we made a clones.
Thank you, Dr. Kaul. Thank you very much.
Thank you so much.
That your lecture has actually created so much of interest. And
Thank you so much. I'm happy. Thank you.
Because it's in the talk of the day. Thank you very much.
Thank you so much. Thank you.
What is next?
Before we proceed to the next lecture, Dr. Nutan Kaushik ma'am has to leave because of some urgent work. So we would like to felicitate her before she moves on. And I request our chair and co-chairperson, Dr. Bandyopadhyay and Dr. Krishnani, to kindly felicitate our distinguished speaker. I may request you to please step down so that from the dais so that we can have a good better photograph together. I request Dr. Nutan Kaushik ma'am to please come forward. Kindly put your hands together. We thanks a lot, ma'am, for intellectually enriching session which you have given. Thanks. Thank you, ma'am. Now we will proceed with our next lecture of Dr. Deepak Ganjiwala.
Now, the next speaker is Dr. Deepak Ganjiwala. He is our professor at Amity Institute of Biotechnology, and he is both an educator and researcher in the field of plant biochemistry and biotechnology. And he has nearly 25 years experiences in higher education, research, and mentoring students. He has established himself as a leading researcher, a pioneer researcher, and made significant contribution in plants secondary metabolism research, mainly essential oil, biosynthesis, and regulation in lemongrass. That's another interesting medicinal plant. He also works on bioprospecting and microbial biotransformation of secondary metabolism. He has published 90 research articles, including research papers, book chapters, and book. He has successfully completed several research projects sponsored by national funding agencies, and in recognition of his achievements, he received the Distinguished Scientist Award from the Society for Plant Research, India 2024. Furthermore, he is the recipient of Israel Government Scholarship, DST Young Scientist Award, and Summer Research Fellowship of the Indian Academy of Sciences, India. So may I invite Dr. Deepak just to give his presentation.
So let me tell you one thing.
Your time is on.
Yeah. Sun is about to set, and when sun is about to set, I'm going to on-play my lecture. That is busy of this particular lesson, which I will be talking on the minute biotransfer of monotherapy and monotherapy in those molecules. So, in that sense, I would like to set the objective of this entire work, and that is the plants are the major source of many bioactive compounds. Plethora of bioactive compounds, secondary metabolites, phytochemicals, natural products are produced by the plant. And these secondary metabolites or phytochemicals, in process, they demonstrated remarkable bioactive properties. And also they are immensely used in commercial, industrial, and many other sectors. That means every aspect of human life, we can say every aspect of the human life is impacted by the secondary metabolites or the phytochemicals, one or in the other way. So our entire work philosophy is that these secondary metabolites, although they are very important for the humans or the industry or the commercial sector, but unfortunately they are produced in extremely low quantity in the plants. And therefore researchers and the scientists have always been looking for alternative approaches or exploring many approaches for enhancing or improving their production. There are several strategies have been applied and tested over the years. Among that, the microbial biotransformation has been emerging as one of the very prominent or excellent tool for the production of valuable secondary metabolites. So this is the abstract of the lecture, but I should not forget that to say on the onset, I would like to thanks our esteemed chairperson, my faculty colleagues, and few remaining my dear students and the co-chair volunteers. Thank you so much to be present. Yeah, till this time. So now coming to the lecture, the monoterpenes and the monoterpene indole alkaloid. What exactly they are? Monoterpenes, as you can see, they are the most significant class of flavor and fragrance compounds. And these monoterpenes, they are also major components of the essential oil. They are mainly biosynthesized in plants via two biochemical pathways. Since, earlier, like up to 1993 or 1996, it was believed that they were mainly biosynthesized via acetate MDF pathway. But in 1993, a new breakthrough has come in the field of this monoterpene biosynthesis. And according to that, monoterpene actually biosynthesizes via MAP pathway, that is the 2C-methyl D-erythritol 4-phosphate pathway, which actually operates in the plastidial compartment. So monoterpenes are exclusively produced from the IPP, which are derived from the MAP pathway. On the other hand, monoterpene indole alkaloid, as the name indicates, that they are the nitrogen containing compounds which are mainly derived from the tryptophan as a precursor, and later on they joins hand with the monoterpene and ultimately or eventually they form the indole alkaloids. So in our work we were interested to biotransform some of the valuable monoterpenes and the indole alkaloids. For your information, I want to say that I would like to bring into your notice that plants have many such pairs of secondary metabolites, such as like geranyl acetate and the geraniol, which are the monoterpenyl acetate and monoterpenol. Similarly, you can have several other pairs which are comprising of alkaloids, flavonoids, or phenolic compound. Among them, the vinblastine to vincristine, which are also very important, significant pair from the point of view of its anti-cancer activity. They are most potent anti-cancer agents available to date. And then the agemelicine and the serpentine. These pairs in the plants, they are interchangeable, and plants converts the part like one side of the pair into the other side with development, and lot of enzymatic machinery are involved in this biotransformation. but it takes some time and the yield at the end will be very low. Therefore, we wanted to perform this biotransformation of agemelicine to serpentine, vinblastine to vincristine, or monoterpenyl acetate into monoterpenol such as geranyl acetate outside the plant using the homologous enzymes from the microbial source, so that we can not only enhance their production, but also we can achieve this biotransformation in a very short time and in a very efficient way. Now this is the pairs. These are the pairs of the secondary metabolites or the phytochemicals on which we have worked to convert them into the more value-added compound. You can see in the plant, geranyl acetate is converted into geraniol by the action of enzyme esterase. So we were interested to see the same enzyme esterase from the microbial source, or we wanted to transform geranyl acetate into geraniol using the microbes or the whole microbes. Then we have the vinblastine, which is also biotransformed in the plant with the help of enzyme peroxidase. To date, it has been reported that peroxidases are involved in the biotransformation of vinblastine and vincristine. Though vinblastine also have anti-cancer and anti-angiogenic activity, but vincristine is considered more potent anti-cancer agent than the vinblastine. And the reason behind is that the methyl group has been converted into the peroxide group into the vincristine, that makes it more potent compared to the vinblastine. Similarly, agemelicine can be converted into the serpentine, and these two compounds, they are used in the cerebrovascular diseases. They are actually vasodilator and antihypertensive, so they have tremendous applications in the pharmaceutical sector. So far, the major sources of the production of these compounds are: number one, for the geraniol, geraniol is mainly extracted from the plants such as the lemongrass or many varieties of the Cymbopogon species. And also there are chemical synthetic methods are available in which geraniol is biosynthesized chemically from the myrcene or from the pyrolysis of the beta-pinene. Similarly, levendulol is also one of the very interesting and important monoterpenol which is used in the spices and the pheromones. So nowadays, this levendulol is demanded for the production of pheromones, and particularly pheromones are used in the insecticide and the pesticides to kill the pest or the insects which are harmful for some crops. Then this levendulol is chemically synthesized from the photocatalytic hydrogenolysis from carbon, and it is also very recently produced in the Saccharomyces cerevisiae by the metabolic engineering or the biotechnological process, in which the scientists have incorporated or introduced the genetic machinery or enzymatic machinery in the Saccharomyces cerevisiae and made it to produce the levendulol. So this is very recent paper I have come across, and these are recent developments in the areas of the production of these compounds. Similarly, vinblastine and agemelicine, most of their production is dependent on the plant. But as I told you, the amount or the quantity or the yield of these secondary metabolites in the plants are extremely low. Therefore, it would not be feasible to rely on the plant for their large scale production, and considering their increasing demand in the world market for several reasons. So therefore, we were interested in their biotransformation. Microbial biotransformation are the processes which rely mainly on the microbes and their enzymes, which we can also call biocatalysts. These processes are very efficient, rapid, and specific and substrate specific, and can be completed in 1 or 2 steps. The most important aspect of the microbial biotransformation is that we can produce optically pure isomer from the substrate. That means the problem of racemic mixture can also be resolved using the microbial biotransformation. So that gives microbial biotransformation upper hand compared to other technologies or other method or other approaches available, particularly chemical synthesis, which has been considered highly detrimental and hazardous for the environment. So therefore, we have opted the microbial biotransformation to convert vinblastine into vincristine and agemelicin into serpentine. And we got very exciting results, and we could able to establish the biotransformation system which can efficiently convert up to 95% of the substrate into the product. So with a very good recovery rate. However, the yield and all these things has yet to be worked out to enhance the yield to certain level. But yes, we could be able to establish the biotransformation system which can be further scaled up for the large scale production of these valuable pharmaceutical compounds. So microbial biotransformation in our laboratory we have performed, and the entire methodology has been summarized in this slide. You can see here the bacteria or the fungus or the endophytes which we have isolated from the particular plant were incubated with the substrate, and then the biotransformation reaction took place in the liquid media, and then we extracted the product of biotransformation. After that, the product of biotransformation was analyzed first by TLC or some what you can say preliminary techniques or separation methods, and after that they have been analyzed by the GCMS or LCMS to confirm the presence of the product. The LCMS, GCMS and all this technology or techniques have clearly indicated or detected the presence of the product in the biotransformation reaction mixture, indicating that these microbes or bacteria are capable of biotransforming these compounds. So following were the potential microbes we have tested for the biotransformation: bacteria, fungus and the endophyte. Among endophyte, particularly Fusarium equisetae, which we have isolated from the Catharanthus roseus for the first time and tested for its ability to biotransform vinblastine into vincristine. And we have found that this endophyte was very efficient in metabolizing or biotransforming vinblastine into vincristine. Because as you know that endophytes are those microbes which live with the plant, and while they live with the plant, they also learn all the processes which plants do or all the biochemical pathway which plants operate. So they actually becomes very smart and they start mimicking the plant biosynthetic pathway. So as Catharanthus roseus, which is the major source of the alkaloid like vinblastine and vincristine, which is also called vinca alkaloid because of the vinca rosea. So this we thought that the endophytes or the bacteria or the microbes which are living with the Catharanthus roseus plant will also be able to convert the vinblastine into vincristine. And as we expected, we isolated the Fusarium equisetae and it was characterized morphologically and molecular level, and then it was identified as Fusarium equisetae. Similarly, in case of the bacteria, we have tested several bacteria: Pseudomonas aeruginosa, Pseudomonas fluorescens, and Pseudomonas putida. Among them, the Pseudomonas aeruginosa and fluorescens have been found highly effective in biotransforming the vinblastine and vincristine as well as geranyl acetate into other compounds. So before we use them into the microbial system, this is the characterization of the Fusarium equisetae. First, we did the morphological characterization, and after that, we went for the ITS gene sequencing, which confirmed that it is Fusarium equisetae. So before we perform the biotransformation, it is very important to check the tolerance level of the microbes to that particular substrate. And that is done with the help of substrate toxicity test. So we have grown the bacteria and the Fusarium equisitae in a medium containing the substrate to varying concentration. And then from here we have established the varying the concentration to which the microbes can easily grow in the system. So substrate toxicity test has provided us Fusarium equisitae, Pseudomonas aeruginosa, and Pseudomonas fluorescens as the prominent or prospective microbes capable of doing biotransformation. So we continued with this 3 microorganisms to develop the biotransformation system. So when this is the biotransformation system which we develop for the geranyl okay, for the geranyl acetate. In the biotransformation system, we have taken the liquid media, that is the nutrient broth, and then it was inoculated with the 48 hours microbial culture, and the substrate was added to this mixture, and the reaction was allowed to take place over the next 7 days at 37 degrees centigrade and 150 rpm. The geranyl acetate, after the incubation period, produces geraniol and nerol as the major product at pH 7, while levandulol was produced at pH 5 and 9. The production of the geraniol, nerol, and other levandulol and other metabolites were confirmed by the GC-MS, and we have seen that almost 67% geraniol and 19% nerol were produced within 5 days of the incubation period, and 55 to 59% of the levandulol was produced at pH 5 and 9 on the 7th day of the incubation. Then here this indicates the course of the biotransformation of the geranyl acetate. You can see on the day 1 only geranyl acetate was there in the reaction mixture. Later on day 3, the geranyl acetate was first conformed into the amcimine, and later on day 5, geraniol was first appeared in the reaction mixture, and later on the level of geranyl acetate has gone down and the level of geraniol has gone up. So that means all the geraniol was converted into the geraniol, which can be seen with the accompanying decrease in the amount of geranyl acetate with the increase in the amount of geraniol. Also here I want to tell you one more interesting thing, that we have also isolated the enzyme which is responsible for converting geranyl acetate into the geraniol, and we have extracted the enzyme esterase, and the activity measured of the esterase enzyme is clearly indicated as that the activity of the esterase enzyme, which is non-specific and the specific, both activity we have measured, which indicated that it was collateral with the geranyl acetate biotransformation. As the rate of biotransformation is increasing, the activity of esterase enzyme is also increasing during the 7-day incubation period. So this has clearly indicated the role of esterase enzyme in this biotransformation process. From the pattern of the biotransformation product, we could also establish the biochemical pathways underlying this biotransformation, which is indicated in this slide, and we have proposed 3 or 4 routes which is leading to the different products of the biotransformation. So the biotransformation mechanism has also been elucidated from this work. Next, we have established the biotransformation system for the conversion of vinblastine and agemelicine using the bacteria two Pseudomonas species, and this work has been published in the scientific report. And here we have incubated the substrate, definite amount of the substrate, 1.2 molar vinblastine and 2.8 molar azmelecine was incubated with the bacterial culture, and again incubated for the 8 days. And from 1 to 8 days, we have seen that the vinblastine and azmelecine were converted respectively into the vincristine and serpentine. And their production in the reaction mixture was confirmed by the LCMS analysis. This is the biotransformation system which has been established with the help of the Fusarium equisetae. And here also Fusarium equisetae were able to convert vinblastine into the vincristine, again confirmed by the LCMS analysis. And then we went for the optimization of the biotransformation system, and with respect to the substrate concentration and the temperature as well as the pH. So the optimization has revealed that the biotransformation system 1, which utilizes 0.6 millimolar vinblastine and the agitation speed of 150 RPM, as well as the microorganism culture concentration was 150 microliter of 8.2 into 10 to the power 5. This was the number of microbes, that is the CFU count, and the spore count for the Fusarium equisetae. So this concentration of the culture and the substrate has produced maximum conversion rate of 92% in case of the Fusarium equisetae on the day 8, while in case of the Pseudomonas fluorescens, 95% of the vinblastine was converted itself on the day 1. So this indicated the efficiency of the bacteria and the endophyte in biotransforming the given substrate. Further, we have also established the biotransformation system using immobilized enzyme. So using immobilized whole cells. So we have immobilized the whole cells in order to check whether they are showing more efficiency than the free cells. So cells were immobilized using the sodium alginate entrapment method, and the results of the free cells and immobilized cells are compared here. We can see that the biotransformation system established with the help of Fusarium equisetae, which is referred as FEBS, Pseudomonas aeruginosa referred as PABS, and Pseudomonas fluorescens referred as PFBS, clearly indicated that the rate of biotransformation using free cells was comparatively more than the immobilized cells. However, upon immobilization of the cells, the total number of spores were a little bit lesser than the free cells because of the effect or impact of the material used for the immobilization. Therefore, overall efficiency of the immobilized versus the free cells were almost identical. During the biotransformation of the vinblastine and azmelecine, we have encountered one challenge, and that was of the structural complexity in the azmelecine, and because of that, the azmelecine could not be converted completely into the serpentine, and the yield of the serpentine produced from the azmelecine was very low, and that is because of the positive charge present on the nitrogen atom, which is actually responsible for precipitating along with proteins that cannot be avoided. So we are working in this direction to enhance or to resolve this problem and to enhance the production of the serpentine from the azmelecine. Now, as I told you, vinblastine and vincristine, they are anti-cancer and anti-angiogenic compounds, anti-mitotic compound because they work as anti-cancer agent by attacking on the microtubule. So we have checked the anti-angiogenic potential of the biotransformation via produced vincristine through chem assay, and the chem assay clearly indicated that the vincristine produced from the biotransformation has shown equal activity to that of the pure vincristine. However, in this direction, more studies are needed to standardize the doses of the vinblastine on different types of cancer cell lines. So these are the further challenges and prospects. I'm just concluding. So this work clearly indicated the potential of the microbes such as 2 Pseudomonas species and the 1 endophyte Fusarium equisetae in biotransformation of pharmaceutically important compounds, vinblastine, vincristine, as well as the geranyl acetate into geraniol and levandulol. Vinblastine and vincristine, they are very well known anti-cancer agents, while the levandulol and geraniol, they are highly used in the flavor, fragrance, perfumery, cosmetics, and deodorant industry. Similarly, levandulol is also very important compound in the spices and fragrance industry. Apart from that, its importance in the pheromones is rapidly increasing. So therefore, this work is very timely and has provided or underpinned several breakthroughs in this particular direction to produce these compounds via microbial biotransformation. And in future, we will be surely working on the operating cost of this biotransformation system and to scale up this biotransformation system to produce these compounds at industrial level. So with this thing, I would stop the presentation here itself. Thank you so much, and I would be delighted to take your 1 or 2 questions. Thank you so much. Rest question you can come and ask me in the lab.
Good evening, sir. Was nice hearing you. My question is, during the biotransformation of the endophytic fungus, biotransferring vinblastine to vincristine, have you come across any the plausible pathway, metabolic pathway? Because in endophytic fungi or any endophytic microbe, there is a very the knowledge of the biosynthetic pathways are very less known. So have you come across any of those possible pathways or any alternative pathway that is the fungus is following?
Yeah, I understood what you want to ask actually. See this, please sit down. The vinca alkaloid biosynthesis and their regulations have been abundantly investigated in the Catharanthus rosea as well as in Rauwolfia serpentia. At the same time, their biosynthesis in endophytic fungus have also been investigated. So the biosynthetic pathway, which starts from the erythrose 4-phosphate and phospholipid pyruvate till the synthesis of the vinblastine and the serpentine, is very well known, elucidated, and is very well known. Not only that, all the genes which encode the enzymes of all 35 steps takes place from phosphoenol pyruvate and erythrose 4-phosphate up to this level, and all 35 enzymes and their genes have been isolated, purified, and characterized. Not only that, very recently, Zhao et al. Have incorporated all these 32 genes into the E. coli and made it to produce vinblastine and vincristine. However, they could not make the bacteria to produce vinblastine and vincristine. Rather, they were successful to produce vindoline and catharanthine. And then that vindoline and catharanthine has been extracted from the bacteria, and later on, chemically they were synthesized into the vinblastine and vincristine. So the biochemical pathways leading to vinblastine, vincristine, and all these things is very well known, even in the endophyte as well. Because the endophyte which I talked today, Fusarium equisetae, there are several other endophytes in which people have already studied the biosynthesis of the vinca alkaloids.
Thank you. Very nicely and lucidly, your presentation, and you made the difficult subject so that people like us who are sitting over here, not knowing the different pathways, only knowing that only knowing the microbial transformation can actually help us in terms of deriving or getting different types of pharmaceutical and also fragrances.
Yes.
So the fragrance part I was missing.
Yeah,
But
surely.
yeah.
Nowadays actually industry is also looking for different types of monoterpene derivatives. This monoterpenes, they impart different types of aroma like lavender or hydrocarbons. Yes, hydrocarbons. Yeah. So many derivatives of the hydrocarbon and the different types of olfactorial properties, which is good for the human use, even in the aromatherapy and aroma industry, which is completely based on the monoterpenes and different types of derivatives. So demand of these compounds is increasing in the flavor, fragrance, even in perfume industry. And as we know that now the trend has been also shifting from synthetic chemical to the naturally produced synthesis. So in the larger population, this shifting has also dictated researchers to look into different types of monoterpene derivatives which can be more useful from the industrial point of view.
Dr. Deepak, there are some terpenes and terpenoids and alkaloids. They are bactericidal. So when you use this as a precursor for bacteria, there's a possibility that, because it depends on the initial concentration which you are using it. Am I right?
Yeah.
So it may be bactericidal for the bacteria, the culture which you are using it. Am I right? Maybe you can throw some light on that.
Yeah. Yes, because many alkaloids and terpenoids, they are known for their remarkable antimicrobial properties. And even in our case also, when we started screening of the microbe, some microbes could not tolerate the concentration of the substrate like alkaloid or the viral acetate because they are very potent antimicrobial agents. But in due course, there are some microbes which can tolerate this concentration of this substrate. So this screening process itself a very tedious process and lengthy process. So while we screen them, we apply different types of concentration to which they can tolerate it. And sometimes the microbes can themselves evolve some mechanism to evade this kind of compound as well. So this is what you can say, it is a long process of screening. And if we are not successful in getting the microbes through screening, then researchers can produce recombinant bacteria by introducing antibacterial resistant gene in them. Yes.
Thank you. Thank you very much.
Thank you.
Thanks a lot, sir. With this talk, we conclude our session on plant and agriculture biotechnology. I really thanks all our speakers because they gave a very intellectually enriching session it was. Now, to conclude the session, we request our chair and co-chairperson to kindly felicitate our distinguished speaker, Dr. Deepak, and I may request to please step down, our chairpersons, kindly step down from the dais so that we may have a better photograph with our speaker. I will ask Dr. Deepak sir to kindly move forward. Kindly put your hands together. Now I will also request Dr.
Dr. Ranjit, please come.
Yes, Dr. Ranjit and Dr. Shagnika, we can have a group photograph.
Yeah, why not?
Chairpersons also, I will request Sumedha ma'am and Nidhi Chaudhary ma'am, Dr. Girish Sharma, Dr. Shagnika, Dr. Nidhi Srivastav to come forward and this is a small token, our gratitude, our thanks to our distinguished chairpersons for guiding us throughout this session. We thank you a lot, sir. Thanks for taking out time and joining us for this session.
घर पे देखना है
Now I will request everyone to proceed to I2 for the high tea, which we have missed in between. Sorry, sir. So I request you all to proceed to I2 for it.